Burlingame, California. Meeting's called to order. We're going to start with the Pledge of Allegiance. If you are able, please stand, put your right hand on your heart, face the flag. Scott, would you lead us? Thank you.
I pledge allegiance to the flag of the United States of America and to the republic for which it stands,
Nation, God, indivisible, with liberty and justice. Thank you. Scott, we need to start with the taking of the roll.
Thank you. E.L. Masri, Kim Barrett,
Present.
Dan Bernal, George Bloom. Here.
Present.
Linda Boxer, John Carruthers,
Present.
Marguerite Casillas,
Present.
Judy Chow,
Present.
Leandra Clark Harvey,
Present.
Shannon Dahl,
Present.
Deborah Dees, Anne Marie Juliet.
Present. Here.
Sorry, just for the record, my mic keeps coming off. One more time. Isabel Derone.
Here.
Thank you. Mark Fisher Colts. Here. Mark Fisher Colbury. Here. Elena Flowers.
Present.
Judy Gasson.
Here.
Jeff Golden for Shlomo Melman.
Present.
Vito Imbasciani.
Present.
Rich Lahara. Pat Levitt.
Here.
Hala Madanat.
Here.
Linda Malkus.
Here.
Carolyn Meltzer.
Present.
Chris Miaskowski.
Present.
Adriana Padillo.
Here.
Joe Panetta.
Here.
Marv Southern.
Here.
Shauna Stark.
Here.
Carol Watson. Yael White.
Here.
Kevin Zhu. Keith Yamamoto.
Here.
Thank you very much, Mr. Chair. We have a quorum.
Thank you. We will proceed to items 3 and 4 taken together, which are discussions of personnel items related to the chair and the vice chair. And for this, I will hand over this part of the meeting to the chairs of the governance subcommittee, Judy Gasson and Pat Levitt.
Thank you very much, Vito. At this point in time, Scott, would you please read the language that takes us into closed session?
Yes, so for these two agenda items, the board will be adjourning to closed session for a discussion of personnel related to items 3 and 4 of this agenda pursuant to Health and Safety Code Section 125290.30, subdivision F3 capital D. So for those board members on the Zoom, you should see a queue on your screen to join the breakout room. Please click join and we will see you all there momentarily. And for those in the room, everyone will clear out except for Raphael and myself.
And Maria Bonneville is excused for this part of the meeting. Thank you.
Okay. Now.
On agenda item number 3, which is an open session consideration regarding the first item, which involves the vice chair.
And
And-
Judy has an announcement to make.
Okay, and I was just about to pass the gavel metaphorically to Judy.
Thank you, Vito. And I want to report that no action was taken in the closed session.
Thank you, Judy. Continue.
Thank you very much. We're now considering, and I will now entertain a motion to approve the 3% merit increase for the vice chair. I would like to request a motion, please.
So moved. So moved.
And a-
Second.
And is there any debate on this matter?
Kim Barrett.
Kim? Did you raise your hand?
No, sorry, I was speaking as to who made the motion.
Oh, I'm sorry. I'm
May
sorry.
I just clarify that for the maker and second of the motion, that this is 3% retroactive to July 1st, 2025.
Precisely.
Yes.
Thank you. Okay, hearing no further discussion, may we take the roll call, please?
Comment? Doesn't appear so. Kim Barrett,
Aye.
George Blumenthal,
Yes.
John Carruthers,
Yes.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Deborah Dees,
Yes.
Anne Marie Deliege,
Yes.
Isabel Derone,
Yes.
Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes.
Judy Gasson,
Yes.
Jeff Golden for Shlomo Melmed,
Yes.
Vito Imbasciani,
Yes.
Pat Levitt,
Yes.
Hala Madanat,
Yes.
Linda Malkus,
Yes.
Carolyn Meltzer,
Yes.
Chris Miaskowski.
Yes.
Adriana Padilla,
Yes.
Joe Panetta.
Yes.
Excuse me, Joe Panetta, thank you. Marv Southern,
Yes.
Shauna Stark,
Yes.
Carol Watson,
Yes.
Yael White,
Yes.
and Keith Yamamoto.
Yes.
Thank you very much. Then that motion carries. Judy?
Thank you very much. We will now take up the chair's request and turn it over to Vito for a short presentation to open this item.
Microphone, please.
Thank you, Judy, and thank you, Pat, and members of the governance subcommittee and the full board for considering the request. The first thought that you probably should have had when you read my memo, what are the responsibilities of the chair and how would they be covered if this request were to be honored? So I should tell you that with Scott's help and direction, I scoured Proposition 14 and the internal governance policy of the ICOC and came up with 14 responsibilities of the chair and have divided them effectively in half so that I would maintain responsibility and focus on 6 of them, which I have listed in the memo. That includes managing the ICOC meeting agendas and workflow, and that includes weekly meetings with our CEO and president, with our chief counsel, and with board governance. Ex officio member and participating member of all board subcommittees, oversee the annual audit and the budget creation for the office of the chair, and to appoint advisory task forces as needed. The last time that was done was over 2 years ago with the creation of the Neuro Task Force. That leaves 8 other activities, 2 of which, numbers 4 and 7, are really rare. And I am assured in conversations with the governance subcommittee and with our president and CEO that devolving these responsibilities in part to members of the CIRM team will not overly burden anyone. And I say that with experience because the various teams, for example, the communications team that does so much work in data collection and patient selection, and other activities related to the creation of the annual audit, they're already doing this work. And I participate in an editorial and a guidance way and giving final approval. The same with the preparation of the budget and preparing for the bond sales twice a year. So I'm assured that this is covered, and I guess if you have any questions, this would be a good time for that.
Not
I think
hearing, oh,
Yeah, Judy, we do not yet have a motion on the table.
I was going to.
Yes.
I will entertain a motion to approve the chair's request.
So moved.
Second, please.
Thank you, Chris.
And is there any discussion upon this motion regarding the mechanics and the merits of this proposal? I'm not hearing anything. Scott, are there hands raised?
There are not. So
Okay.
I think we can move to
For a vote then.
public comment. Let us pause. Doesn't appear there's any public comment. Kim Barrett,
Aye.
George Blumenthal,
Yes.
Maria Bonneville,
Yes.
John Carruthers,
Yes.
Marguerite Casillas,
Yes.
Judy Chow,
Aye.
Leandra Clark Harvey,
Aye.
Shannon Dahl,
Yes.
Deborah Dees.
Yes.
Thank you. Anne Marie Deliege.
Yes.
Isabel Derone,
Yes.
Mark Fisher Colbury.
Yes.
Elena Flowers,
Yes.
Judy Gasson,
Yes?
Jeff Golden,
Yes.
Pat Levitt,
Yes.
Hala Madinat,
Yes.
Linda Malkus,
Yes.
Carolyn Meltzer,
Aye.
Chris Miaskowski.
Yes.
Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Marv Southard,
Yes.
Shauna Stark.
Yes.
Carol Watson,
Yes.
Yael White,
Yes.
and Keith Yamamoto.
Yes.
Thank you very much. Judy, that motion carried.
Thank you all. Back to the chair.
Oh, thanks, Judy, and thanks, Pat, too, for guidance in all of this. Okay, we are now moving on happily to agenda item number 6, a discussion regarding the process CIRM is using for preference setting. I'm going to introduce our president and CEO, Jonathan Thomas, to lead the discussion and make necessary introductions. Thank you, JT.
Thank you, Mr. Chair. Just very briefly, as the board well knows, the concept plans adopted for the new PDEV and CLIN 2 programs under the SAF include preferences as an important part of the process. There have been a number of questions about how the preferences have been working, what's appropriate, what's not, et cetera. And we're pleased that now that we have 2 full rounds of pre-submission analysis for PDEV and qualification analysis for CLIN in the books, that we now have data that makes it appropriate for us to give the board and the public a full accounting of retrospectively how the preferences have played out over this initial phase of implementation under the SAF. And to do that, we have our esteemed Chief Science Officer, Rosa Canet Aviles, who's going to give the board a presentation on this topic in detail. Rosa?
Thank you, JT. And thank you, Mr. Chairman, Madam Vice Chair, distinguished members of the board and the public. I'm happy to come here with an update on the funding preferences. Just before I get started, can anyone tell me if you can see the presentation well?
We can.
Okay, wonderful. So as JT was saying, over the last year, we introduced these preferences that to help translate the public investment that we are given through Proposition 14 into what we are expecting to be the real patient impact within our remaining runaway. So what we are bringing today to the board are the results of the first cycles, to give us all a chance, as JT was saying, to pause and to assess how this is working in practice. And I would like to start with the end of what we intend to achieve today, so that there is a focus in today's discussion. Today is about the intent, is about the early signals that we've seen and what we are learning, not about final conclusions. So the goal at the end of today is to discuss and learn from the board, from you, what types of data you would like to see at our next March ICOC meeting, to be able to make decisions moving forward. So with that, I'm just going to move into today's layout of the presentation. The overview has the context and objectives of why are we doing this, where are we, the guiding principles that moved us, and we will go through those guiding principles, and then the results of the first cycles for the three programs, the preclinical development and the clinical development, CLIN2, and then this four, which also has had one cycle where preferences were applied, and the review is happening soon. And then closing, what are the next steps? And then we can open it for the discussion. So this slide is meant to ground the rest of today's discussion in the reality, in the operating reality that we are all working within. So we are working within Proposition 14, which significantly expanded our mandate, both in terms of scientific ambition, but also in the expectations around patient impact. Cell and gene therapies have moved forward. When we started in 2004, it was a very different field that we are now, right? So in particularly, it reinforced priorities around diseases of the central nervous system, and made access and affordability an explicit part of our responsibility, not something to consider downstream. At the same time, our funding remains finite and time-bound. So we have a defined remaining runway, and the amount of strong applications scientifically that we receive consistently is much larger than what we could support. And this happens to all funding agencies, so it's not a unique problem to CIRM. So this is not a reflection of declining quality, it's a reflection of the demanding outpacing capacity that we can take. So taken together, these two realities, the finite funds and the expanded mandates and the increased amount of applications, create a central tension that we are trying to manage. So the expanded expectations for impact within fixed resources and finite time frame, and ultimately the goal is to translate all of this investment into therapies that reach patients. But therapies that reach patients that could not otherwise be reached in California. So to that end, the board approved the strategic framework and the impact goals as the roadmap to achieve that back in September of 2024. So preferences are the mechanism by which we introduce to help navigate this challenge. And the rest of the presentation builds from this reality, how we attempted to operationalize all of this, what are we seeing from the initial cycles, and what else do we need to bring to the board so that you can make informed decisions before considering any next steps. So starting from this context and reality, the practical question is, how do we move forward from many strong applications to a portfolio that has the highest likelihood of delivering real patient impact within our lifetime? This slide shows the role of the preferences, the role that they are intended to play. And it's not a substitute of scientific review, but a way to focus limited resources towards applications that are most aligned with our impact goals. On the left, we start with a very high volume of applications. Many of these are scientifically strong, but they vary in terms of readiness, alignment with the statutory mandates, and the likelihood of translating into therapies within the time frame that we have. The stars here in this slide represent applications that most strongly align with our guiding principles. It doesn't mean that the others are not scientifically strong, it's just that the ones with the stars are more aligned with those guiding principles. And importantly, the absence of the star doesn't mean that they are not strong scientifically, as I was saying. So the preferences help enrich the pool by bringing forward applications that are more likely to contribute to near or midterm impact given our constraints. So that's what the preference role has been. And that enriched pool then moves into scientific and technical review, where the merit, the rigor, and the readiness are evaluated in depth. The key point is that preferences shape which applications rise for review, while the peer review determines which of those will get funded. Now, I want to pause here and acknowledge that what we've been hearing, because this feedback is an important part of why we are having today's discussions. Also, I want to make sure that everybody knows that there is a memo as well that contains all of this presentation and a lot of the details that we had been asked through the science subcommittee in terms of data. So if you want to refer to that while I'm presenting, that's also available. So some of the concerns reflect uncertainty about how the preferences or this pre-submission or / qualification process interacts with the scientific review, and whether the proposals that didn't advance were fully considered on scientific grounds. Others reflect questions about whether preferences might unintentionally favor certain modalities or pathways, or whether progression within the CIRM portfolio is being appropriately recognized. We have also heard a desire for more clarity around how qualification scoring and preferences and intent fit together, and what the preferences are designed to do versus what they are not designed to do. More importantly, a lot of this feedback is less about disagreement with the goals and more about understanding the logic. In other words, how the pieces fit together, how decisions were sequenced, and how this ultimately connects to delivering therapies for patients. And that feedback has helped us understand that we need to be clearer and more explicit in how we communicate the rationale behind these decisions, and in how we show the link between prioritization decisions and the outcomes of patient impact. And also about the fact that if we want to make an impact with the constraints that we have of time and funding, we need to make hard choices, and that we need to be more clear about it, and to make very clear what the choices are in alignment with the board, obviously. Taking all of that together, the context that we are operating in, the role of the preference are meant to play, and the feedback we've been hearing, this brings us to what we are trying to accomplish in today's discussion. We have 3 objectives. I just want to make sure that the objective today is not to debate individual outcomes or to make decisions about changing preferences. That's going to happen in March. I just want to make this very clear. The objectives today are about reviewing the rationale for these preferences. How did we set them up? How do they connect to our proposition for teen mandate and to the strategy that the board approved back in September of 24, and to the practical constraints that we are working within? That's a very important point, and we hope that this will be clear after today's presentation. The second is that we want to share what we are learning from the first funding cycles. These are signals, not conclusions, but they will help us understand whether the approach is functioning in the way that it was intended. And part of our questions today to the board are there any specific analysis of these first funding cycles that you would like to see more granularity in the data so you can understand this better at the March meeting to make your decisions? So that would be the second part. And the last objective today is that we want to use the committee's perspective to identify what portfolio analysis levels are needed before we come back in March to have a more substantive discussion about these next steps. So the second and the third are kind of go together. Let me see, what did I do? So now we are going to go with that framing before getting into any specific preferences or scoring mechanics. Is important that we go back and make explicit what were the principles that guided how these preferences were developed in the first place, because that doesn't seem, we didn't do a good job explaining this. So I'm going to spend quite a bit of time in this slide. These principles are not a theory. They are basically the backbone on how we've designed all our programs, but specifically we are talking now about the preclinical development and the Clean 2. And they reflect the realities also of what we've learned over the years of funding programs that either did or didn't translate into therapies that reach patients. The first principle is that they need to offer potential for transformative clinical impact. What do we mean by clinical impact? We are not using that as a synonym for scientific novelty or disease importance. In the context of preclinical and clinical programs, clinical impact means the realistic potential for a therapy to meaningfully change outcomes for patients in terms of efficacy, durability, safety, or burden of care. Compared to what exists or is potentially coming down the pipeline. This principle forces us to ask, is the science interesting? It doesn't force us to ask, is the science interesting? What it's asking is, if this works, does it actually matter for patients? The second guiding principle is that it addresses bottlenecks to access, affordability, and translational feasibility. This principle reflects one of the most important lessons embedded in CIRM's strategy. Early decisions that we make affect downstream access. In both the preclinical and the Clean 2, we have seen that choices around modality, delivery, manufacturing strategy, and the clinical implementation can make the difference between a therapy that's technically successful and that one that is actually usable at scale. So this principle is not about pricing policy or reimbursement debates. It's about whether the proposed approach acknowledges and begins to address known bottlenecks to access. The third principle is that it fills critical funding gaps and advances CIRM's statutory mandates. This is a very important principle. So CIRM's role is not to fund everything, is to fund what others cannot or will not, which was part of the reason, our raison d'etre when we were established in 2004. This principle reflects the reality that some diseases, modalities, or stages of development are persistently underfunded by federal agencies or by the private sector, despite their potential impact and importance. In preclinical development, this shows up in how we think about enabling translational work that could otherwise stall. In the Clean 2 program, it shows up in supporting trials that carry scientific or operational risk, but that align strongly with CIRM's mission. This principle also explicitly incorporates Prop 14 mandates, including CNS and pluripotent stem cell approaches, as part of our prioritization logic, not as exclusions, but as areas that CIRM has a statutory obligation to lead. The fourth principle is that it can realistically achieve key regulatory and development paths within CIRM's finite runway. This principle is one of the hardest and also a very important one. It doesn't mean that ambitious science is discouraged. It means that we have to be honest about timelines, dependencies, and what CIRM funding can reasonably enable within the years that we have left. In the Clean 2 program, this is why readiness, regulatory clarity, and operational feasibility matter so much. in preclinical development in the PDEP program is why we focus on whether a program is positioned to make a meaningful inflection, such as enabling an IND. This principle, and rather than accumulating more exploratory data, for example. And this principle protects both CIRM and applicants from investing heavily in programs that, even if scientifically sound, are unlikely to reach meaningful milestones within CIRM's lifetime. The fifth principle is that it addresses diseases affecting Californians. As a public funded agency, CIRM has a responsibility to ensure that its portfolio reflects diseases and conditions that meaningfully affect Californians. This doesn't mean funding only common diseases, nor does it exclude rare diseases. What it does is requires us to be thoughtful about the overall balance of the portfolio and the populations ultimately served in that. And that's something also that as part of the portfolio that we bring in March, if you have questions about this, we want to make sure that we incorporate them. And the last principle is diversification of CIRM's active award portfolio. This final principle is particularly important given the concerns that have been raised about preferences becoming restrictive. Diversification is a guardrail. It exists to prevent overconcentration in a single disease area or modality or development pathway, even if that area is popular or well represented among applications. In practical terms, this principle is why novelty, underrepresented disease areas, and differentiation relative to the active portfolio are explicitly considered in the PDEP and CLIN2 scoring. It is also why preferences are not binary gates. And it ensures that genuinely novel or non-obvious approaches, including outliers, have a pathway forward rather than being crowded out by programs that simply align with the most common preferences. With those guiding principles in mind, I want to show now how they were translated into practical program-specific preference rubrics. And in the next slides, I will walk through both the PDEP and the CLIN2 slide rubrics, which were intentionally designed as complementary tools at different points along the translational pathway. In both cases, the key thing to keep in mind here is that these are alignment and qualification tools, not substitutes of scientific peer review. So the PDEP program is intended to enrich CIRM's clinical pipeline. Compared to the existing portfolio, it should advance state-of-the-art therapeutic technologies that address a broad range of diseases affecting Californians. The first line here reflects alignment with core areas where CIRM has a clear role and responsibility, either because of our statute or because of downstream access and feasibility considerations. Applicants in the first line of preference received up to 3 points if their project aligns with at least one of these areas, either pluripotent stem cell-derived therapies, diseases of the CNS, or in vivo gene therapies. Importantly, in this case, this is not an additive point. Meeting one criterion is enough. And if you meet two, you get the same. This was designed deliberately to avoid stacking modality bias or forcing projects to fit multiple boxes. The next set of preferences captures readiness and translational momentum. These include non-viral acid nucleic acid delivery, evidence of early FDA engagement through a pre-IND or Interact meeting, and progression from earlier CIRM programs such as DISC2 or the TRAN1. These signals help us understand where a project sits on the development path and whether CIRM funding is likely to be catalytic at this stage. Again, these preferences are not requirements. And you get a point for each one of these if you get this, if you click that box. Underrepresented disease areas, this category is about portfolio balance. We explicitly look at how represented a disease area already is within CIRM's active awards and give modest additional weight to areas that are underrepresented. The intent here is diversification, not prioritization of rarity or exclusion of well-represented diseases. And the last one is the novelty criteria, which exists specifically to prevent preferences from becoming exclusionary and to ensure that strong outlier ideas still have a way to move forward, even when they don't score points or multiple preferences. At the same time, it's important to be explicit that no single preference operates in isolation. Projects still need to touch other aspects of program intent, and preferences inevitably will involve trade-offs. Setting preferences means making choices about what the portfolio is optimized to do, and that doesn't exclude some things, even when the science is strong. The next one is the Clean 2 rubric, and this is at the qualification. So this slide summarizes what we've used for the Clean 2 qualification rubric, which is used as an internal screening tool by the review team to determine which applications, and this is for applications, the PDEP was for pre-submissions, so there is the difference between the PDEP and the Clean 2. So the review team determines which applications move forward based on this rubric to the grants working group. It's not a scientific scoring rubric, and it doesn't replace peer review. Each item here reflects a discrete preference designed to assess alignment with the guiding principles that I mentioned a few slides ago. The first set of preferences includes pluripotent stem cell-derived therapies and diseases of the CNS, which are both explicit Prop 14 priorities. And this is aligned with the PDEP preferences as well. In vivo gene therapy is included here indirectly, responding to a statutory mandate because of its potential for scalable delivery and broader patient access. Non-viral genetic therapies are included to recognize emerging approaches that may reduce safety or manufacturing constraints. We also account for accelerated regulatory designations such as RMAT, Breakthrough, or Fast Track, because these materially change development timelines and signal regulatory alignment. Progression from earlier CIRM awards reflects a stewardship of prior investments, and California-based organizations reflect the taxpayer-funded nature of the program. Finally, pivotal trials receive additional weight because they represent the fastest and most direct path to potential licensure and patient impact. Something that is not here, but that, well, I'll talk about this later. So with that framework in mind, we'll show how the results from the first two cycles of PDEP and Clean 2 illustrate how these preferences played out in practice. I'm going to start with the PDEP and the first cycle. This slide summarizes how the preferences framework played in the PDEP first cycle. We received a total of 168 pre-submissions, reflecting what I was saying at the very beginning, a very strong demand and high volume of applications. Using the pre-submission rubric, we invited 33 applications forward for full application and review, which is roughly the top 20%. What's important here is the separation signal. Among the invited group, 97% met 3 to 4 of the stated preferences, whereas 42% of the non-invited proposals met 0 or 1 preferences. That tells us the rubric was functioning as intended, not excluding ideas arbitrarily, but helping distinguish which proposals were most aligned with the priorities that the board set. And during the science subcommittee presentations, we had questions about how the preferences were distributed by the applications. Those details are actually in the memo. So if you are thinking about this, please refer to the memo for those details. Now, of those 33 invited to full application and review, 12 were ultimately funded awards. And we see several consistent themes here. There was a clear increase in proposals with a stronger translational readiness, innovation, and scalability. This is all aligned with the guiding principles. More than half represented progressions of prior CIRM-funded programs, and the funded set is more enriched for disease areas that have been underrepresented in our active portfolio. Taken together, these cycles suggest that the preferences are not narrowing the science, but rebalancing the preclinical portfolio across modality, disease area, and Prop 14 priorities in a way that's intentional and aligned with the strategy that the board approved. This slide is going to now show the data for the second cycle of the preclinical development. An important caveat here is that these are pre-submission results only. We have not yet completed a full application review or grants working group assessment. What we see, however, is that the same pattern from the first cycle is holding. We received 126 pre-submissions and invited 23 for full application, representing approximately 18% of the total. Importantly, 100% of the invited applications met 3 to 5 preferences, while about a third of uninvited applications met 0 to 1 preferences. So the signal here remains very clean. 2 additional points respond directly to questions that we've been hearing. First, the invited pool spans broader disease areas, including more cancer and hematology programs than in cycle 1. And second, nearly 40% of invited applications could represent progressions within the existing CIRM portfolio if funded, which is exactly what we intended as well. So while final funding decisions are still ahead, the takeaway, the preliminary takeaway is that the preference framework continues to do what it was designed to do, which is to identify the strongest, most aligned programs early, while allowing the portfolio to broaden across disease areas where the science is ready. I'm having trouble today with the clicks. Okay. So now I'm going to turn into the Clean 2 Cycle 1. We received 23 total applications, all of which were ranked using the preference points as part of the qualification. From those, 7 advanced to full review and 4 were ultimately funded. A few things are worth highlighting here. First, all the 7 applications that advanced to full review met 3 to 4 preference points. So the framework that we put together with the preferences clearly distinguished the top tier. All 7 targeted CNS indications, which is consistent with one of the Prop 14 priorities. There's been some questions here about whether if we remove CNS, it could change the pool of what advanced. We've done that, and we could bring this in March. It didn't change the nature. It actually changed in terms of oncology. It changed which application of oncology moved forward, but it didn't change the picture because all the CNS indications are also clicking other preference criteria in the qualification process. The second conclusion here is that 6 of the 7 involved either pluripotent stem cell-derived therapies or in vivo genetic therapies, and 4 already had advanced regulatory designations, which speaks to development maturity. Looking at the funded programs, all four have feasible delivery paths aligned with our access and affordability strategy. As you know, we've developed a rubric. We have consultants evaluating this, and that's a very strong part of our review process as well. And three represent progressions of prior CIRM funded programs. And this tells us that the system is reinforcing both innovation and disciplined portfolio advancement. The key takeaway is that the system has been working directionally, there you go, as intended, and if elevated programs that are aligned with the statutory, it has elevated programs that align with the statutory priorities, clinical readiness, and delivery feasibility. That said, this first cycle also made clear that the relative weighing of preference matters, and that's where refinement might be needed to ensure we are not over-concentrated signals as the portfolio grows. This is the second cycle. This slide shows where we are for the second cycle, recognizing that these are interim results pending the application review subcommittee approval of the grants working group review that happened 2 days ago. We received 21 applications, all of which were ranked using the same preference-based framework. From those seven advanced to full review, and every advancing application scored again 3 to 4 preference points, which is consistent with what we saw in the first cycle. In terms of composition, 4 targeted CNS, and those 5 represent progression from the existing CIRM portfolio, and 5 are either in vivo genetic therapies or pluripotent stem cell-derived therapies, again, reflecting strong alignment with the preferences that the board has set. And the final funding recommendations will follow the application review subcommittee approval, which I believe this one is the March one, but Scott will confirm. So stepping back, when we look across the preclinical development and the PDEP and the CLEAN-2 first cycle together, the overall pattern is consistent and directionally aligned with what the board asked us to do when we set the strategy. Awards in both programs reflect the innovation, readiness, access and affordability, and CNS criteria that were explicitly laid out in the program announcement, not informally, but operationally through the qualification and ranking process. The applications that advanced were largely CIRM portfolio progressions, but importantly, they were not advancing because they were legacy projects. They advanced because they also met multiple stated preferences, same with the CNS. As a result, the portfolio now includes more feasible and scalable modalities, which strengthens downstream access and affordability potential without sacrificing scientific rigor. That said, I just want to make sure that we are clear about the caveats here. These are early signals, and it's too soon to assess portfolio level impact. What we can say at this stage is that the system is behaving directionally as intended. I'm now going to move into the DISC-4 results. And as you all recall, the DISC-4 preferences were set different than what we did for PDEP and CLEAN-2. This was done through the Neuro Task Force Science Subcommittee. And it's a brief reminder that the staff proposed alternating preference cycles for these 4 with neuro every other year. And the board approved a neurological disease as the preference for fiscal year 25-26 for the DISC-4 cycle. The program was open to all comers, but there was this preference. And when we say neurological disease, it was in the wide understanding, including neurodegenerative, neuropsychiatric, and neurodevelopmental diseases. This slide shows how that alternating structure is intended to work over time. Some cycles are driven by Neuro Task Force Science Subcommittee identified needs, such as the first round that we had in neuropsychiatric or neurodevelopmental priorities, while others reflect the board set preferences. And it was Prop 14 Neuro wide understanding of the preference that the board set back this year. So crucially, every cycle remains open to all comers, but the preference does determine, what we will mostly be reviewing. This structure gives the boards a repeatable, transparent way to set direction year over year, while still preserving scientific breadth and flexibility. So now I'm going to show what were the preference scorings for these four in this year. These are how we operationalize the preferences consistent with what was approved in the program announcement. So the neurological disease preference carried a very large weight. You can see here that if you didn't apply neuro, you could still make it through, but it was hard. So the neurological disease carried 36% of the total scoring weight, and that was intentional and transparent, which reflected the board decision to clearly signal a priority for this cycle while still keeping the program open to all applicants. Importantly, the preference alignment was not the sole driver of the ranking. 64% of the score came from scientific substance, which was represented by relevance to human disease biology, cross-disciplinary and system-level approaches, and innovation in stem cell or genetic research. In other words, as I was saying, projects still had to be strong science first, and the preference weight did not replace scientific merit, but it shaped which strong projects rose to the top when trade-offs were necessary. This structure is doing what it was designed to do, which is to translate the priorities that the board gave us without collapsing these four into a single disease program or excluding high-quality work outside of the preference area. Now I'm going to show what did we get as results of setting these preferences in this first cycle. We received 138 pre-submissions, and 86% aligned with the neuro preference. That tells us that the signal was clearly heard by the community. From that pool, 24 applications were invited to full application and review, and importantly, 100% of those invited met the neuro preference, but they span a broad range of neurological diseases and represent a wide diversity of mechanisms and scientific approaches. And that part of, we will be happy in March to provide a download of granular download of that data. So what we are seeing here is not convergence on a single disease or modality, it's enrichment within a priority area while preserving scientific breadth. Funding decisions, as I mentioned earlier, are still pending, and we will have a review in 2026 in February. But at this stage, the preference is doing exactly what the board designed to do, which is to shape the pool without narrowing the science. So I'm just going to close with 2 slides. Stepping back, these are the signals that we are seeing from preference setting across the programs. First, applications that advanced to grants working group review consistently met multiple preferences. This tells us that the rubric is doing a real triage and is not advancing projects on a single attribute alone. The second is that in Clean 2, we see high proportion of CNS projects funded, which is consistent with the stated preference and confirms that when we weigh a priority, it shows up clearly at the outcome level. But it also shows that for those CNS projects, they are hitting other multiple preferences. So they are not coming in, is signaling that the field is ready for CNS projects. In contrast, for PDEP, it shows a much wider spread across disease areas and modalities, which reflects both the earlier stage of the science and the way that preferences were designed to guide, not constrain the portfolio. And finally, across both programs, a large fraction of funded projects are CIRM portfolio progressions, over 50% in the case of the PDEP program, and roughly 75% in the Clean 2, suggesting that what we've set up is reinforcing continuity while still applying directional filters. Importantly, these are early signals and not final conclusions. And this presentation, what we are intending to do is tee up key questions for the board. And in March, we would bring portfolio analysis and options informed by your guidance. Now, based on what we've shown today, what are we going to bring in March? The first thing is we will propose paths in March to move forward that will be informed by A, what sort of preferences could the board like to see, and B, where in the process does the board want to see the preferences? So those are two very important things, right? The sort of preferences and where in the process do we have these preferences? In order to have that discussion, we will provide portfolio analysis that allows us to visualize how preferences are shaping what's moving forward in terms of the portfolio. During our pre-calls that we had with the science subcommittee, we got feedback on those analyses already. And that those are reflected here. First, we will bring illustrative portfolio cuts that show how certain funded projects map across innovation, pipeline progression, and disease focus. That includes concrete examples of what we are categorizing as innovation, how projects advance through successive stages of CIRM funding, and how diseases are represented across recent cycles. Second, we will present a clear picture of the current and emerging portfolio, looking at both active awards and recent application cycles, and broken down by disease area, modality, stage of development, and how preferences in the first cycle have been shaping the pipeline. With all of this, the proposed analysis intent is to move the conversation from individual applications to portfolio level effects. The second thing and please let us know if there is anything there that we are missing from the science subcommittee feedback or from questions that you might have now. The second thing that we plan on bringing are options for where preferences could operate in the funding process. For example, should we have them at pre-submission, at qualification, or later? At the application review subcommittee as a programmatic considerations, and what each option implies for scientific review, for transparency, and board oversight. This will be framed as options, not recommendations, to support the discussion in March. And with this, I would like to thank you all, the board and the public, for the attention and all the questions as well that have helped us shape these presentations, which we hope that it brought some clarity around the process and the stage that we are in as we are trying to reach our goals. And with this, I would like to bring it back to back to you, Mr. Chairman.
Thank you, Rosa. Thanks for the comprehensive presentation. Also, I want to thank you for the care and the breadth and the scope of that memo that you and Vice President for Review, Gil Sembrano, supplied the board with. It was a great document to help frame the discussion. So I expect sort of broad and robust discussion from board members before we go to the public. So I'd like to open the floor to discussion on this item.
Judy Chow,
This is not an action item.
Judy Chow has her name raised as well.
Okay, okay,
Hand
thank you.
raised.
Let's start with Judy then, and George will follow.
Again, I will echo Vito's comments. Very nice presentation. And I particularly also appreciate the memo because that really helped to frame the whole discussion. I do think when we look at the PDAF and also Clean 2, and I am a little bit surprised about we didn't mention anything about the manufacturability. If you look at today's cell gene therapy, the biggest bottleneck to become a feasible product, no matter for individual patient or the broader patient population, the biggest issue is there. And I know CIRM, we have put in quite a bit of effort and also investment in some sense to this area. But I do think when we set up the criteria, particularly in the later phase, it should have some manufacturability to make sure it truly can benefit the patient and also guarantee that robustness. Again, I heard you, Rosa, you've been saying those words, right? Scalability, the manufacturability, but I feel we need to formalize that because this is so important.
Very good point, Dr. Chow. And the manufacturability is taken into account in the review criteria of the applications. What I was trying to convey in the guiding principles was the access and affordability. But you're absolutely right that manufacturability is essential in terms of being able to access and afford, right, in the access and affordability. So thank you very much for that point.
Thank you, Judy. George?
Yes, thank you so much, Rosa. I think that was an excellent presentation, and I think this is a really important topic for the board, particularly in view of the volume of applications that we are getting and will continue to get in the future. And it'll be an important tool in helping us distinguish which applications can move forward. So I think that's really good. I also think it's really good that this affords the opportunity of greater transparency to those who are applying for grants to have a better idea of what the criteria that the board will be using in terms of decision making. My question, and this may really be a question for our discussion in March, is how do you envision us approving a final list of preferences for each offering? Is this something that would be done at a board level, or will this be done at a CIRM level before each offering?
As always, a very, very good question, Dr. Blumenthal. And before I answer that question, I just want to mention that we have heard loud and clear if we did not do a good job in communicating and being transparent, and we are really making a big change. That's why the memo, but also it's going to be in the program announcements, and we'll be very clear in our webinars as well. So I think more than two ways I would answer the question. The first one is for March, there could be the decisions that I think would help us as a team from the board could be where could the board like to set the preferences? Where is the placement? Should the preferences continue to be applied upstream at the pre-submission or qualification? Or do we want the preferences to be shifted later in the process? And that's one question. The second one is, does the board have, when we've looked at the principles, I think it's not so much about the preferences, it's about the principles that we are applying to set those preferences. Is there a disagreement with those principles from the board? Because those are the core, I mean, the way we translate those principles to the preferences it's kind of it's more scientific, doesn't really have a lot of leeway, but it's the principles that we all need to be adhering to and in agreement. So perhaps a discussion could be, are these the principles that we all adhere to? And that could help us, because the preferences are more of a mathematic translation of those principles. Thank you, Dr. Blumenthal.
Thank you, Rosa and George. Before Pat asks a question, Scott has an issue of clarification.
Sure, just for a process point, for those members of the public participating on the Zoom, want to remind you that if you wish to make public comment, please refer to the process described on the agenda, because it's slightly different from how we've invited participation in the past. We won't call on you from the Zoom, but instead through the dial-in information that is at the top of the agenda posted on our website. So wanted to give that reminder while the board continues its discussion. Thank you, Vito.
Scott, could I say something to finish Dr. Blumenthal's question that I forgot and Dr. Patel just reminded me?
Sure.
So I should have specified that the preferences are approved by the board when we present our concepts. So I think that if there is a revision, or if the board desires a change in the preferences, we would have to come back with a revised or amended concept for the board to discuss that. So that could be the procedure. But I think what I was saying earlier on is the guiding principles is what we all need to adhere to. And if there are discussions about that, then that deems a discussion about the preferences for the programs.
Okay, thank you. Next will be Pat, followed by Maria, and then Kim. Keith.
So thanks, Rosa. The great thing about preferences is that you don't know until you institute them whether they have unintended consequences, and then if they do, then you can adapt and adjust to them, so they're not super glued. So I've two comments. One is about the guiding principles. If you read the guiding principles, number one, offer potential for transformative clinical impact, and number four, can realistically achieve key regulatory developmental path within CIRM's finite runway. Those are really highly related to each other. They go hand in hand. There's a microphone. You can't hear me? Can you hear me?
Yeah, I can hear you.
Well, it's pretty clear they're trying to silence me.
Always.
So 1 and 4 are basically, I'm not saying they're identical, but they're basically very complementary. You can't have potential transformative clinical impact without realistically achieving key regulatory, et cetera, et cetera. And then you have 3 and 6, fill critical funding gaps and advance statutory mandates. There's no issues with CIRM's advancing statutory mandates. That's our charge. Filling critical funding gaps and diversify CIRM's active award portfolio, basically the same thing. So in fact, I think we have 3 guiding principles. One is about transformative clinical impact. The second is about bottlenecks, access, and affordability, extremely important. And the third is about debating where we are now in terms of runway, about diversifying the portfolio. What does that mean? What does that have an impact on in terms of those opportunities that then may be more advanced, but yet are continuing to be part of our portfolio? So that'll be maybe a conversation for March. I don't want to belabor that. The other is that there's a circular argument that if you have preferences and you use those preferences to evaluate a subset of grants going forward, of course, those grants are going to meet the preferences because you use the preferences to make the decisions, right? So it's always a circular argument. The question is whether the preferences need to be re-examined. So for me, there's issues around 2 things. One is what would seem to be a bias against cell-based therapies, which can't get scored. It doesn't get a point. And then the other is non-viral nucleic acid delivery, which I think is a very important platform. In fact, we're going to hear later about a new initiative, which is very exciting, that is essentially non-viral nucleic acid delivery in most ways. And so we have two components now that are basically pushing non-viral delivery when we know that there are still major opportunities for viral delivery. The vast majority of approved FDA therapies are viral delivery. Folks are working hard in this area as well to modify so that there's greater success, just like they are in non-viral deliveries, which is very exciting. So I'm suggesting that CIRM invest in non-viral delivery platforms, right? But we have two components now that are favoring that. And basically, if you're doing viral delivery, you're behind the eight ball. So this is all to say, I'll stop there, that I think revisiting the preferences based on the initial outcomes, I think is worth some significant period of time during the March meeting. We're not going to have enough time in this meeting to go through all this, but I just raised those 2 points. Thanks.
Great, Maria. Then
As something Rosa mentioned about if we change preferences, it's the change in the concept plan, and that has to come back to the board. I want to understand a little bit, and Scott or JT, is the team the team will make recommendations in March for perhaps what preferences might look like. If there's more conversation at that meeting, will we be able to change? I assume there would be a concept plan that would come in order to allow for timing, because I think that's why we wanted to have this conversation earlier, because of the concept plans and whether or not June is soon enough. I'd heard some conversation around that. So I just want to make sure that if there are changes to the concept plan that are decided at the March meeting, they're outside of recommendations that have come from the team, that all of that timing works.
So yes, that's the idea.
Which part? I'm sorry. Which part?
So you're asking whether you'd be able to make adjustments to the concept plan at the March meeting as opposed to waiting till June?
Yeah.
That's sort of the crux of the question. So I think Rosa, if you want to step in here, but I think the object here is to get the new preferences in place as soon as we possibly can. And if that can be done in March, that's
Okay.
the preferable way to go.
They would need to be in March because of the timeline for the different programs that are coming in. So the idea was to have kind of a blanket approval, like if we have a discussion around preferences and process, because the second thing is, where do preferences come in? And I think that there are some trade-offs about having the preferences earlier or later, right? But so that could be one question. And the other could be, if we discuss the preferences, and you can see PDEP and CLEAN2 are aligned in preferences, then if we were going to do this every year, right? Revise the preferences. That could be like a blanket approval that could then flow into the concept. We don't have to present the whole concept again. It's just that board has set these preferences for this upcoming year based on this data, and that's now translated into the concepts. That's how I was conceiving this, and I don't know if Scott and you, JT, agree with that.
No, that's entirely in line with what I just said. So thank you, Rosa.
And the second thing I wanted to mention, and just in conversations that we've had a lot of conversations about this, but something that I think we miss is, and it's something that Pat mentioned, what are the outcomes we want? Because that really drives what our preferences are. And so in a clear alignment around an outcome, or the outcomes that we believe are important, is really the conversation we need to get to. And I think then everyone is more clear on what the preferences mean and why they're there. So thank you, Pat, for bringing that up.
that wasn't a rhetorical question, right? Okay. Next is Kim, followed by Keith.
Rosa, thank you so much for a really careful exposition of where we've been and where we are right now. I appreciate that. I also agree with you that the response that we've received from the community illustrates that we have to do more to communicate, and I'm glad that that's in the plans. I think we can all agree that in a perfect world, we would have enough resources to fund every project that was scientifically meritorious. And but clearly neither both in amount nor time frame, we don't have that. So I personally believe that we have a need for preferences and that they need to be applied early, because otherwise we lose the efficiency of the review process and the ability to focus on the proposals that are most aligned with our priorities. So the request I have for March, you know, I'm a scientist, I like data. In the scientific literature, in my own field and many others, there's been a revolution over the last 5 to 10 years to be much more explicit about individual data points. And so with the understanding that averages and proportions can hide details of the data information. So I would really hope in March that you can provide us with the actual distribution of preference points for the full application pool and those that were moved forward for funding. I know it's expedient in a short memo to summarize those data, but I would, for one, would like to see the actual data of every single proposal, how many points it received, and what the distribution was for both the ones that were put forward and the ones that were not reviewed.
We will be happy to provide that. For Clean 2, it's going to be easy because there's like 44 total applications that came in. For PDEP, it will be complex, and for these 4, but we will figure out a way to do it, and if not, it will be presented, and it needs to be anonymized, but it will be presented as in the form of a memo. I want to make sure that that's possible as well. Yeah.
I'm happy to talk to you offline about ways that the data might be depicted, and it certainly can be anonymized. I'm only looking for the distribution here.
That'll be very helpful. Thank you so much, Dr. Barrett.
Thanks, Kim and Rosa. Keith? And then Shan.
Thanks, Rosa, for the presentation, and thank you for, more importantly, thank you for all the work that you have done in framing this idea of finding ways to pre-assess whether applications qualify, given the fact that we're going to be faced actually with an increasing load of applications. Having said all that, I'm actually deeply concerned about this program. Now, you might ask why I didn't raise this earlier. We heard that Pat and I and maybe others around the table around the Science Committee and heard this presented before, and we happily passed it on to be presented at this meeting. But having thought more about it, and also read and thought about the public comments that we've received, I'm deeply concerned, and my concerns start with 3 of the 6 guiding principles. Pat talked a bit about the guiding principles, but I have concerns about 3 of them. One is the need to offer the potential for clinical impact. I think for fundamental basic studies, that's a hard call. And the consequence of that, of enforcing that, is taking a rather short-term perspective on the ways that we think about an application, and that would necessarily lead to more support for more conservative work, work that is clear, that is kind of high on the feasibility score, but maybe not very deeply considering its potential impact, even if it's not apparent at the point of application. The second guiding principle I'm concerned about is that whether the work addresses bottlenecks in access, affordability, and feasibility. These three also really drive conservative thinking and evaluation of, or choosing to evaluate conservative applications, things where it is immediately apparent. And here, affordability is an assessment of affordability is a particularly flawed criterion. All new technologies are not affordable at the outset. And I'll just, everybody remembers, but I'll just say it that the first human genome sequence cost $3 billion. And that cost has come down 10 million fold. So trying to make a guess about affordability at the outset is flawed. Third, achieving regulatory and developmental pathway within CIRM's finite runway. And the problem I have with this one is that science can't be actually scaled or assessed or evaluated on the basis of whether it fits into this artificial timeline that we are stuck with, if you want to put it that way. First of all, we don't know that we're stuck with that, with an end or true endpoint. But more importantly, there's really nothing wrong with getting work started and doing something significant when you can, and then making it apparent to the rest of the world, that is to say, other funders, that there's value in being able to extend that work. So if it's not done inside of the CIRM pathway, but CIRM getting something started that ends up being highly significant, it's not a bad thing. So I'm concerned about half of the guiding principles on which this idea is founded. Secondly, I think we learned something, learned more than what you presented, Rosa, in those first two cycles of experimenting with this program. As Pat said, it's no surprise that the applications that move forward for review and were ultimately funded fit the pathway, the program well, because of the circularity that Pat talked about. But what wasn't considered, so the fact that the applications that went forward for review and then those that were ultimately funded scored well on the preference list that was constructed is not very surprising. What wasn't considered, but I think we heard about in some of the public comments, maybe all of them, is were there applications that were highly meritorious that did not score well and therefore were triaged? That's not so good if that happens. And I think what we heard in the public comments is that it did happen. And we don't want to begin to build a system which is sort of rigged in the sense that highly meritorious proposals don't even go forward to review. And I think that's the situation that we're in. So considering that denominator, as it were, not just the ones that went forward and therefore qualified very well, but also asking ourselves much more difficult question of whether there are applications that are highly meritorious that are being eliminated because of the way that the system is set up. And I think we need to be very careful and thoughtful about that. And so my final point is that we heard in the public comments that at least in the view of certain applicants, there were highly qualified proposals that were put forth and subjected to this experiment. I think it's fine that we did the experiment, subjected to the experiment that were triaged. And I think that those people need our attention, and that we need to go back and look at those and consider them. And it's not limited to those that we heard from. So there's going to be a fair amount of work in doing that. But I think that we owe that to the applicants. It's fine to do an experiment. It's important to do experiments. We're scientists. But we need to be able to look at the outcomes in ways that we can assure ourselves and the applicants that we're being as equitable and just about making these judgments as we can.
Thanks, Keith, for those remarks.
Dr. Imbasciani, could I respond or make some clarifications?
Yeah, okay, Rosa, sure.
Thank you. Thank you, Dr. Yamamoto. As always, very excellent points and assessments. I could just like to take the opportunity to make a few clarifications, if I may. So with the, and hopefully I took good notes. The first point was about clinical impact is in terms of the guiding principles that is hard for fundamental basic studies, but these programs, what we were talking about in terms of the guiding principles are not funding fundamental basic studies. We are talking about the preclinical development, the clinical development programs. So when we say clinical impact, we are not asking to predict downstream value of discovery science. We are asking about the development stage of proposals that we are evaluating. I just wanted to make that as a clarification. The second point in terms of access and affordability, we were not talking about price prediction. It's about feasibility of the delivery and the scalability, and as Dr. Chow also mentioned, manufacturability and avoidable barriers as the downstream deliverability of these therapies, right? There was something else about there was a third point, was about the one I wanted to make. There was one point, there's a third point, and I can't remember because I didn't take notes. I was trying to think about the response, but the last one is about applications that are deemed highly meritorious. Well, without peer review, we don't know if they are highly meritorious, right? So the preferences are classifying based on objective qualification or preference criteria that if you click it, then you get to be able to figure out if there is a highly meritorious. Now, are there guardrails? So that's one of the questions for the board. Is there an explicit pathway for high novelty or transformative applications that have not met the standard preferences? And I know I'm getting myself into a bit of a potentially trouble here, but are there guardrails that the board would like us to have to figure out if there are some that don't meet the preferences that would need to go to review? I mean, we cannot make everybody happy. And I think that's one of the problems that we've had. We've not been clear about what we would not be funding. And I think as a board of 35 members, there is a decision that needs to be made because we cannot fund it all, right? So I just wanted to make those points. But I really appreciate the thinking because that's the kind of discussion that we should have had. Thank you.
I have a list of 5 speakers, but Keith, I'm going to offer you the opportunity to rebut if you would like.
Well, let me keep this brief. Thanks, Rosa. I appreciate that. And I'm not saying that every applicant who feels that this triage system has eliminated the potential to compete with what every applicant considers their work to be highly meritorious. So I'm not saying that denominator, as I refer to it, is a firm number, that everybody that was triaged and feels that it wasn't fair should have been funded. But I would argue that many, based on the letters that we received and reading them fairly carefully, it seemed apparent to me that what the authors of the letters were arguing is that the ways that the preferences were structured, and maybe even the point systems assigned to the preferences were structured, eliminated their work kind of in a preemptive way that didn't even allow the evaluation to go forward. And I think that's what we need to be cautious about. I think it's, don't misunderstand. I think that's complicated and difficult to do, but I think we owe it to applicants not to construct a system that preemptively eliminates them for reasons that aren't directly associated with the quality of the application.
Thank you, Keith. So here's what I have for a list. Judy Gasson, Isabel, Shannon Dahl, Marguerite Casillas, and Yael, but I'm afraid we lost Judy.
Mhm.
We did. Okay, so Isabel, you're next then.
Great.
Thank you.
Thank you. Rosa, thank you very much for the refresher course. We've been discussing this a lot, and I'm very pleased to hear and learn more so I can keep chewing on this. As a patient advocate for oncology, a cancer survivor myself, and a concerned voice, loud voice for the Latino community where cancer is the second leading cause of death, I always want to support new, emerging, and advanced cancer research with the hopes that it's ultimately going to reach and serve my underserved community. As 40% of the California population, major contributors to the economic backbone of this state, and taxpayers to boot, the Latino community is as anxious as anyone to be healthy and have their children survive. So I'm very empathetic to the letters that poured in from our cancer researchers. Thanks, Keith, for your wrap-up. I appreciated that as well. Have we, CIRM, been fair in funding across diseases as we try to struggle to meet our goals and use taxpayer funds wisely? I can't help but think and remember and even cry with and for the numbers of parents who have stood before us in these prior public hearings and shared private struggles and fear and hopes and research, most often for their children suffering from these rare diseases. How often I wanted to fund them all, to give them hope, to let them know we hear their pain, and yet after listening to the rationale from my most learned scientific colleagues, have chosen to say no. For those researchers who wrote us, we did not choose not to honor and respect your work. It's very critical. For any reason, we voted, I think, our best intentions, our concerns, knowledge, and of course, from guidance from experts in the field. So I thank Rosa once again for her refresher course. But rest assured, at least from my perspective, nothing is set in stone, and consistently reviewing ourselves and how we determine decisions comes back to us time and time again. And I know that the people I sit with here today are always well-intentioned with the best for community health, but also for great science. So know that we have heard the concerns from the oncology community, and once again, we will look inward to see if and how we change if it's once again required. So thank you very much for sending in all of your letters, for your concerns, for your engagement. And may I ask you to use that force and that concern in I think, engaging all members of our community in communities in California so that we can see equity and diversity in care and treatment and research. Thank you.
Thank you, Isabel. Shannon.
Thank you, Rosa, for a great summary and for also setting the stage of guiding us to ask for what we should be thinking about for the discussion in March so we can all continue to percolate on the topic. One of the things that stuck out to me about the portfolio, we've heard about different diseases, and I'm going to set that aside for now, and instead think about different stages of development. You know, earlier stage, there's often more innovation in modality. And when we get to later stage, it becomes more about feasibility. Can we get to commercialization? Are we going to really serve the patients of California by delivering something over the finish line? And so as we think about preferences, I wonder if it might be helpful to consider different preferences at different stages. And I know they are slightly different, but even perhaps separating the PDEV and the clinical more so to align with different principles that resonate for different stages of development. And then with respect to information that might help us consider how that would pan out, I was struck, especially in the clinical group, in our last meeting that we did end up with almost too few applications. And so we did have a funding level of one that was below the typical funding line. And so looking at how those preferences are set up at that stage to ensure that we're thinking about maybe more about clinical impact there and what's feasible to get all the way to the funding line. And yeah, just considering different stages for different preferences. Thank you.
Thank you, Shannon. Marguerite Casillas?
Hi, well, I'm really heartened by the conversation here today. Oh, and again, want to thank you, Rosa, for your presentation. It's great to hear that, you know, people are really thinking carefully about this system that was possibly, you know, with good intention, but constructed in a way that is now eliminating some good science. And I'm really thinking about the you know, the comms team and one of the key audiences being scientists in industry and us wanting to feel that they're confident in CIRM programs and proud of California and supported by CIRM. And so I think we saw in those letters that there are definitely some questions there. I don't know how the earlier preferences might have been communicated to those folks. Sounds like there's webinars and other things, but I think it's a point if you know, we make changes or refinements that we really need to be transparent and probably over communicate on what's happening there. That's my comment.
Thank you, Marguerite. Yael, wait.
I'm debating. Anyway, okay.
So are we all.
And I also want to thank Rosa for her presentation. And well, I think obviously we want to have more meritorious applicants, and we want to be transparent with our decisions, and we want to have great communication, and we can't fund it all. I feel like you also can't be surprised when you're applying for an application for a grant to be funded and you don't qualify with all the determined expectations of what we're looking for. So again, there's always room for reevaluation on our part of, do we need to be more inclusive? Have we overlooked something? Have we not? But if the guidelines are stated accurately, I don't know, I feel like you can't be disappointed. Well, you could always be disappointed, but it shouldn't be, I guess, a big surprise. And maybe you need to reevaluate the scope of your research, or, you know, it's a two-way street. It's my opinion.
I'm sorry, we're having a little logistical discussion here that involves lunch and we have some, and we have speakers that have hard stops that must get back to their respective institutions at a certain time. So did we solve that? Okay. All right. I'm really sorry for that interruption. Yael, thank you for your remarks. For your remarks, I'm going to have to go back on the transcript but thank you. I don't see any other hands, but oh, I do. Yes.
I want to get back to Rosa's request, thanking her for her presentation, and I want to agree with Kim Barrett. I think if we're going to apply preferences, we have to do it at the beginning of the process. Otherwise, we don't deal with the huge number of grants that we receive. I, like Kim, like data, and I agreed with her suggestion that we need to see not only the application of the preferences to the grants that got chose, but I want to see the preference applications to the ones that didn't move forward. And that may address some of Keith's comments. The other thing, I'm going to offer a suggestion to Maria. I just was on the last grants working group for CLIN. And what occurred to me is Keith was talking about access and affordability. I was astounded by the applications and how they responded to our criteria around access and affordability for the clinical projects. Because these projects had received FDA approval to move forward largely to a phase 1. And I think the board would benefit from getting some exemplars. I mean, you and I talked about one that was absolutely outstanding in terms, without divulging any, confidential information, in terms of how they engaged with a company that knows how to do an access and affordability evaluation for a product that's potentially, if it's successful, will move to the clinical arena. And the product was not cheap. And the evaluation from the company, the insurance companies that were looking at these, was really, really interesting. And I'll stop there because I don't know what's going to happen with that application. But I do think it would be, if the board agrees, informative to kind of see our rubric and how some examples are being produced. And some didn't do anything and others did exceptional jobs. So others that were on the grants review recently maybe want to comment on that. So those are my points. I would support, in terms of additional evaluations, what Kim said, and then perhaps at another meeting where there's time to talk about our criteria and how we're seeing responses.
I'll definitely work with the internal team to see what sort of information we can bring back at a high level that illustrates what you're referring to. So thank you.
Thank you, Chris. No other hands. So we can invite members of the, do you want to say something about our time schedule, Scott?
Yes, thank you, Vito.
Yeah.
We have important public comment that we want to get to on this item, as has been already reflected by members of the board. We are running up against some conflicting schedules. Our two presenters for Closer to Cures have hard stops at 12:30 and 1:15. You also have lunch that is constrained, is between 12 and 1:30. This is not an action item. My proposal is that once board comment here is, for this first round, is complete, that we table the item, go to the Closer to Cures presentations to you, take lunch, bring it back if necessary to the dais, and then once that is completed, come back to this item to hear public comment and continue any further board discussion. If that sounds amenable to folks.
Any objection to that? Thank you. If there was, I wouldn't know how to handle it.
All right, so for members of the public, please plan to participate via the phone numbers that are indicated on the agenda, as I said earlier. Is there any more board comment at this time? Please feel free to continue with this before we hit pause. All right. So then with that, I would propose that we move to the first presentation and Amy Adams.
Yes, I'm sorry. I'm going to introduce to our Senior Director for Communications, Amy Adams, who's going to make some introductions to two speakers for this novel entry in our agenda. Amy?
This doesn't sound, now it's on. Okay. All right, okay, good. Thank you, everyone. My apologies for interrupting this really fantastic conversation. We do have terrific speakers with hard stops. So consider this us giving you time to think about everything that's happened and ruminate. Okay, I'd like to introduce a new standing agenda item of in-person board meetings. At each meeting, we will invite CIRM awardees to speak in their own words about how CIRM support is making a difference in their work to bring Californians closer to cures. Today, we'll hear from two speakers, and I'm going to introduce them both now. My notes say it so I don't interrupt the flow, but I think it's so that I don't interrupt your lunch. First, we're going to hear from Corey Nicholas. He's the CEO and co-founder of Neurona Therapeutics. He will present results of 2 clinical trials testing stem cell-based therapies for epilepsy. His story shows the power of CIRM funding over many years, with CIRM support for this project starting at the bench and continuing to the results that he'll show you today. Next, after lunch, we'll hear from Crystal Mackel, who's a professor of both pediatrics and medicine at Stanford School of Medicine. She's also the founding director of the Stanford Medicine Center for Cancer Cell Therapy and the director of the Parker Institute for Cancer Immunotherapy at Stanford. She'll discuss results of a clinical trial testing a new approach for treating a deadly form of brain cancer. That involves modifying a patient's own immune stem cells so they'll identify and destroy cancer cells. It's an approach known as CAR-T. So with that, I'm going to invite Corey up.
Hey, hi, everybody. Thank you for the opportunity to come and address the board and to present our latest progress from this program that CIRM has supported for a very long time. So the program was based out of UC San Francisco. It goes back a very long time in the labs of John Rubinstein, Arturo Rosbulla, Arnold Kriegstein. That goes back to 1997 with an initial discovery on a type of brain cell that was really unlike any of the other brain cells we had studied, and had really unique properties as a possible cell therapy for treating neurological disease. I put the timeline here because it shows how long this journey has been. And I joined the effort in 2006, and shortly thereafter, we were the recipients of a generous, one of the first cycles of CIRM awards, the RC1 cycle. That was a comprehensive grant, and that led to a series of discoveries on the understanding of this cell type in the developing brain, its role in disease, and then, of course, how to manufacture and replace it for treating animal models and then eventually human patients. We were able to attract venture capital with these data that were generated through CIRM funding over the years. And then Neurona launched in 2015, and we're now a company of about 100, just over 100 people in South San Francisco, right across from the airport, where we are, and our team has been advancing this through clinical development phases, and now involves hundreds of clinical collaborators around the United States. And I'll show you some of that data that we're seeing, and it has us now at the doorstep of a phase 3 trial with a real chance to take this all the way for patients. I just want to highlight something that Keith mentioned earlier, which is that when we started this, we had no idea what would happen with this science. It was a glimmer in the eye. Nobody knew. I mean, these cells were very interesting, but nobody knew that this would lead all the way to epilepsy. In fact, it was a winding road, and we were thinking about neuropathic pain, Parkinson's disease, Alzheimer's disease, movement disorders, which these cells can also potentially treat in the future. But nobody knew that this would develop, as an epilepsy, potential cure. And so I just want to make the plug that even though this is a closer to cures session, and we need to keep funding those, obviously, let's not forget that we need to fund the basic science to seed the pipeline, because that is the source of innovation. Okay, so we have taken a new candidate called NRTX 1001, which is the first human cell therapy that's ever been advanced into the clinic for the treatment of drug-resistant epilepsy. And we have 2 clinical trials ongoing, each one funded by a different CLIN2 CIRM award. We have other candidates and indications planned, including a very interesting pivot in a population of Alzheimer's disease, which features epileptic types of seizures, where we think the same product can potentially be used. And so we really think of this as, leveraging that investment as a, pipeline and a product opportunity. So, but I'm going to talk about epilepsy. And I think epilepsy is misunderstood as a disease that's a nuisance, that's well managed by medications, rather than the severe quality of life hostage situation that it is for most people with this disease. So there's about 24 anti-seizure medications on the market for epilepsy. And luckily, they work really well at about half the population to control seizures. But unfortunately, for the other half of people, these drugs do not work. And people try them in many combinations for a very long time, and they continue to seize. Now, it's thought that about 600,000 people in the United States suffer from drug-resistant epilepsy. And the most common type of epilepsy in adults starts in a place called the temporal lobe of the brain, which is the hippocampal memory center of the brain. And so these seizures not only are electrical epileptic seizures that impair life, but they affect cognition and memory and much broader quality of life issues. And so, about 500,000 people in the country have this disease and are drug resistant. And it's about 100,000 people in California that are, with drug resistant epilepsy of this type. And really without good options. And so these seizures come in different forms. And if you may know somebody, because it's one of the, I think, 4th most common neurological disease. And so you may know somebody that has had this or have seen a seizure. They can come in any form and they can strike at any time. Sometimes people have them daily. Other times it's maybe just once or twice a month. But even though it's just maybe 1% of the time that someone's seizing in life, the rest of the 99% of the time, they're worried about when that seizure is going to strike. And because of that, it impairs their life. And depending on the frequency, the severity, the life goals of the person, this can be completely disruptive and debilitating. So, these seizures can be staring spells. They can be, involuntary repetitive movements. People can then, of course, fall, lose consciousness, burn themselves on the stove, fall down the stairs. Obviously, they're not driving. They often can't care for loved ones. They're dependent on caregivers that have to stop working. People can walk into the street, they can crash a car. And then, of course, you can have a seizure that doesn't stop, which is called sudden death in epilepsy, which unfortunately, it could be terminal. So what happens to people that are drug resistant? So, for people that have access to insurance, which of course not everybody does, they would see their primary care physician. They would get referred to a neurologist who would prescribe them with anti-seizure medications. And if those seizure medications don't work, and if their neurologist is good and they're not in a healthcare desert, then some of them can get referred to a accredited epilepsy center for a comprehensive workup. There are 24 of these accredited centers in California. Okay, in the Bay Area, these would be UCSF, Stanford, UC Davis, et cetera. Now, they will come to these centers and they'll have imaging and EEG workup to try to map the source of the seizure. And if they can find a focal point, one place where the seizures come from, then some of these patients might be eligible for a lobectomy procedure. And if you can believe it, lobectomy in 2026 is still standard of care for these patients, where we are actually removing pieces of brain, large pieces of brain, or destroying it with a laser. And it's just simply not good enough, because even though it can work for about half of people long-term that go through with those, there's a huge trade-off from the obvious collateral damage of taking out and destroying that part of the brain, mainly permanent memory loss, cognitive impairment, personality changes, and so on. So many people aren't eligible, especially if they have more than one focus, and we'll come to that. But also many people are not interested. And the only other therapy right now that's standard of care is a stimulator called an RNS or DBS device that can be implanted into the brain and stays in the brain, indwelling, forever. Unfortunately, it's a good thing is that it's not destructive to the brain, but it's highly invasive and it hasn't worked to control seizures for most people. It's helpful, it's approved as a reducing seizure burden, but it's not disease modifying. So the unmet need is to find alternatives, ideally alternatives that are not tissue destructive. And so this is why we thought we could do better with a stem cell based regenerative approach. So then what type of brain cell would we want to replace? And we want to replace, as I mentioned earlier, this interesting cell in the brain called a GABAergic interneuron. So a little bit of brain 101, overly simplistic, is that the brain obviously has excitatory neurons that transmit information with electricity. That electricity is propagated down the neuron, and then it excites the next one. And that's how our central nervous system works. But what people don't largely understand is that about a third of the neurons in the brain are not excitatory. They're inhibitory. And they work in between the other neurons. That's why they're called interneurons, to regulate that flow of information to make sure that it's balanced, right? And that keeps seizures in check. And they do this by the neurotransmitter GABA. And in epilepsy, there's not enough of these GABAergic cells around. And so our simple idea, in theory, is to boost them by developing a way to manufacture these interneurons with stem cells and then to administer them into the brain. So how do we do this, right? And it's easier said than done. And as Rosa mentioned earlier, the real secret for how we need to do this is we needed to find a way to scale them and to manufacture them consistently in a way that replicates the biology in our endogenous human brains. And to do this all in a manufacturing setting, in a petri dish, essentially. And so that's not easy in practice, right? And so Neurona took the baton and put a lot of people to work on this challenging problem. And we wanted to do better than cell therapy 1.0 in the past. We didn't want to rely on fetal stem cells. So we turned to a human pluripotent stem cell. In fact, NRTX 1001 is made from a single human embryonic stem cell line, which was true to CIRM's early vision in the early 2000s. And from this one cell line, we can expand unlimited numbers of these interneurons. And then we put them, long story short, we actually take them to a more advanced stage. So not only are they highly pure, but they're actually fully differentiated post-mitotic, and they're frozen. And every time we do this, and we do this actually in South San Francisco here across the airport, we can make billions of these neurons, hundreds of patients of doses for each run. And because they're cryopreserved, they're incredibly consistent. As Rosa mentioned, that is the key. To be a real product for the masses, it has to be consistent. So every time a patient gets a dose, they know they're getting the same safety and potency. Right? And so team in Neurona has done an unbelievable job, unprecedented in this type of fidelity to make this happen. And of course, so then we tested these cells first in animals before the human patients. So we developed a mouse model of this type of epilepsy. And these animals, we can record them with EEGs. They have 20 seizures an hour, so they're severely affected. And then we give a dose of the NRTX 1001 interneurons. And here's a picture 7 months after a single injection, one-time injection of the cells. And every red and green cell is a human NRTX cell that was made from a human stem cell. And you can see that they're beautifully surviving and integrating. And then the next question is, could they reduce seizures? And yes, they do. And so this is a plot of their EEG recordings. Each dot, to the question raised earlier, we want to see individual data points. Each dot is an individual animal, okay? And then you can see over time, 0 is no change, minus 100 is complete seizure freedom. And so over time, only the animals treated with cells in the turquoise, but not the animals treated with the vehicle or the placebo. Only the animals with cells become seizure-free. And it's about half of them that become seizure-free, but overall, most of them respond with about a 90% reduction in their seizures. And this is stable from a single dose. So next, we wanted to look at whether these were just treating the symptomatology or whether these cells were actually disease modifying. So one of the things about this disease in humans and also in mice is that the hippocampal memory center of the brain, where the seizures are thought to come from, it becomes incredibly distorted. And so if you look at the top left image, that's a normal hippocampus. That's the little arrow there is called the dentate gyrus, which is thought to be, where the action happens, where the seizure would start in an epileptic. And you can see in an epileptic mouse on the top right, that becomes completely distorted and blown out. And then as we start to dose the human cells at higher and higher numbers, you can see down the slide that you repair that structure. Right? It becomes less distorted. So these cells are not just treating symptoms, but they're having disease modifying restoration of the structure of that part of the brain. And mice are great, but we also wanted to test safety and delivery in a larger brain. And the only way to do that is to do that in primates. We took a small number of rhesus macaques and we worked with an epilepsy neurosurgeon to figure out how to do this in a way that would be scalable, in a way that all of the neurosurgeons at these 200 accredited centers around the country already know how to do, right? And that something that would be even simpler than what they're doing, which is more invasive lobectomies. And so he devised this approach that's an MRI or CT guided approach to deliver these cells, and you can see the dye, to put them precisely with sub-millimeter precision into this part of the brain where the seizures start. And in the pictures, you can see every one of those red and green cells is a human NRTX intraneuron that's integrating into the primate brain. And this is essentially how we do it in the humans. So after demonstrating safety and doing formal toxicology studies, the FDA let us proceed into a first-in-human clinical trial in adults who have drug-resistant epilepsy of this type in the temporal lobe. And so the key eligibility criteria is they had to be drug-resistant, right? This is a treatment that's not first line. It's for after already people have cycled through medications. And then they come to these centers and they're offered a chance at lobectomy or stimulation or Neurona's clinical trial. And we've seen a lot of patients select our clinical trial because they feel like they want to try something non-destructive first. If it doesn't work, they can always do a more invasive procedure later, right? So that's still on the table for them. But why not try something less invasive first? And so these patients have come in, we give them a single dose of the cells, as I showed. And then because the cells are a single product for all patients, it's called allogeneic or off the shelf, right? And so because it's mismatched, we do give immunosuppression in the first year. We think 1 year is maybe overkill, and we want to test that later to see if we can shorten it. But for this first study, we gave them for 1 year, and then they can come off of that. And then the primary objective here is safety, of course, and the secondary objective is efficacy. So we've now enrolled actually 2 phase 1 trials in these types of patients, where the goal is to enroll 38 total patients across the 2 studies. And this has taken a circuitous route that I'll take you through. The first that was funded by the CLIN2 13355 and these are open label multi-center studies, was a 2-dose escalation in 10 patients, 5 patients at each of the doses. Once we cleared safety, we then expanded these cohorts by 8 patients, 4 patients per dose. And all of these patients have now been treated with the cells. You'll see some of their data in a minute. After this, we added another cohort of 10 people that have the same disease, but without that scar on the MRI called MTS, which is a way to try to get this to more people. And then more recently, we started a second trial that's funded by a CIRM CLIN2 17135 to test the same therapy in people that have this disease on both hemispheres of the brain, where they have no option for lobectomy. So it's even a greater unmet need, right? And these are people that are really in a tough spot. Okay, and in this procedure, they have 2 foci. So we're giving the cells into both sides of the brain in the same procedure. And so to date, we've treated 30 patients. 8 patients have been treated in California. 32 centers across the United States are part of this study. 19 have treated patients, so this is really scalable. 7 centers have treated patients in California. And the data cut that you're going to see here is on the first 26 of these that we presented publicly in Atlanta at the American Epilepsy Society in December. So the primary objective is safety. We haven't seen any serious adverse events attributed to the cells. They've been well tolerated at both doses. We haven't seen any serious surgical complications from the delivery procedure. The delivery procedure is about an hour infusion. The total procedure takes a few hours. And then the patients stay 1 night in the hospital for observation. And I believe almost all of them have gone home the next morning. The only events that we've seen mainly are related to the immunosuppression, and that's in that first year. And so we see nausea, fatigue, but importantly, those have resolved when those people complete the immunosuppression in the first year. And then we haven't seen any cognitive decline. We measured closely a battery of neurocognitive tests, and so their cognition, in fact, has improved in some of these patients. So I want to give a case study on the first 3 patients that were treated with the low dose of NRTX 1001. This was funded by CIRM. And this first patient was actually our 3rd patient. So I'm going to go 3, 2, 1. He was having 3 disabling seizures per month in the 6-month baseline run into the trial. He was managing a scuba store. His passion was deep sea diving. He suddenly had a seizure while he was talking to a customer, blacked out. It kept happening, escalated. And so he had to stop working, quit his dream job. Obviously, he's not driving, started drinking heavily to cope with his decreased quality of life. And when he was offered a chance to enroll, he said yes, but he didn't really understand the magnitude. So he got the single one-time dose of NRTX 1001 into his hippocampus on one side of the brain. And what we've seen is that these seizures have gone away by and large, except for 1 seizure in the 12th month. So after NRTX 1001, his seizures have decreased. Many of these patients are very altruistic. They want to help others. He really likes being part of this study. And he's only had the one. This is, by the way, we didn't interview these patients. We have a firewall. These patients were approached either by the press independently at their universities or by advocacy groups. And so one of the reporters asked him, do you think it was the cells that made a difference? And his quote here is that, he hasn't changed anything else. So what could it be? What else could it be? So the next patient that was featured, I believe, in one of the CIRM annual reports, it was our 2nd patient, a 59-year-old, suffering from 2 disabling seizures a month during the baseline period. So her epilepsy actually started later in life, I believe in her late 40s or early 50s. And it happened suddenly when she was sleeping, and she didn't know about it. Her husband waked her up and said she was foaming at the mouth. She was a pharmacist. Her patients were everything to her, and she had to stop working. She was an avid hiker, backpacking. She had to stop. And so her quality of life was changed and compromised. Wasn't able to see her grandkids. When she did an EEG, it showed 11 seizures over 2 days. Again, she didn't want to have brain destruction, so she said, let's go for it. Let's try the cell therapy. And since then, she's been actually completely seizure-free since the 7th month. And now, you could see the months on the x-axis here. It's been over 3 years from a single dose. She said the delivery procedure was a piece of cake. Fatigue is improved. She's able to hike again, and she's able to get going with her retirement goals, traveling and seeing her grandkids. Yeah. And now for our first patient. And I actually heard the mother of the first patient speak at an advocacy pipeline conference in the Miami area in 2023, 1 year after that patient received their dose of cells. She called it an epilepsy hostage situation for her son. This patient was one of our most severely affected, having 32 seizures per month in the baseline period. Okay, and rather than describe it, I'm going to play a video because I think it could do a much better job than I can at underscoring the magnitude of this.
Everyone, I'm Heather Longo. My son, Austin, is the patient. He's 27 years old. Just a little bit about his situation. Can you hear me okay?
Yes.
Okay. Back in 2016, Austin started to have seizures out of the blue. It was shortly after my husband had passed away from a short battle with cancer. So they thought maybe it was stress induced. But further investigation and MRIs and things, he was diagnosed with mesial temporal lobe epilepsy. And there's a sclerosis area in the right temporal lobe that they believed was a focal point. His neurologist, Dr. Titoff, who is our guardian angel, Austin's seizures were not being controlled with medications, and she was had to keep adding another AED and another AED and up to He's currently on 4 high doses of different AEDs twice a day, and still having breakthrough seizures. So she introduced us to Dr. Babu, Austin's brain surgeon, and he admitted Austin, and they did an internal EEG for about a week. He was in the hospital so that they could see if that indeed was the focal point or if there were other areas of the brain that were causing the seizures, because at this point, he was not able to be left alone. He would have multiple seizures daily. He was working 2 jobs when he started having the seizures and had to quit both jobs in 2021. He just became unable to function and had to rely on me and his little brother to kind of guide him. He was becoming like almost his memory, he wasn't functioning very well. He couldn't remember a lot of things. His quality of life was really suffering. We were seeing how debilitating, and we started calling it an epilepsy hostage situation. So when he was in the hospital for that week for the internal EEG, it revealed that he, before they even started to take away some of the medicine, he was having seizures immediately when they put him on the monitor. So as they decreased the medicines to see what the seizures were, in the end, he had over 200 seizures in a 7-day period. So we met with Dr. Babu, his surgeon, and Dr. Titoff afterwards to go over options for treatment. Dr. Babu said that he could go in and surgically remove that section of the brain that they believe is causing the seizures, but there were risks with that. Another option would be like an RNS, but that really wasn't a likely option. The RNS system just kind of turned us off a little bit. And then him and Dr. Titoff both proposed this clinical trial. Neurona is the company that put out the trial. And what it is 1 dose of stem cells, of human stem cells. They go in through his brain guided by a needle with an MRI machine. And Dr. Babu transplants these cells into that sclerosis area. And then he went home the next day. When the doctor proposed this idea, I, as a mom, was completely terrified. He's the first human being in the world to have it done. And we were very scared, not knowing what the outcome could be, being that it was only done on animals and he would be first. But Austin was familiar with stem cells just from the sports world and things like that and different things. I'm an LPN, so I'm semi-versed in the benefits of stem cells that I've seen throughout my career. But it was still terrifying. And Austin said that he's always been a very selfless guy. I once caught him stealing food to help the homeless man in the park to give him food. And he's very humble and very kind. And so he said, Mom, someone has to be first. If no one was first, then they would never be able to save lives. And he doesn't want anyone else who maybe has had epilepsy for a lifetime. He feels that his epilepsy journey has been semi-brief. Austin's very humble and he minimizes his situation quite a bit. And as a mom, I'm very proud of him. But in the back of my mind was, okay, so if the stem cell doesn't work, then we have a backup to the surgical options. And we just entered it with Dr. Babu, Dr. Beach, Lena, and Dr. Titoff. They're an amazing team. We felt extremely supportive. So we opted to start the trial. And within 3 months post-op, we noticed the intensity of Austin's seizures had improved. The frequency had improved. He was coherent a lot more. He is forming sentences more clear. His balance has been more clear. And so we were a little nervous, like, is this a placebo effect? Or are we just wanting it to work so well? However, it just kept improving. His seizures and the activity and all of that kept improving. they've since been able to decrease some of the, 2 of the AED medications. They decreased the dose and he's doing well. His last seizure was January 27th. And for us, that's very amazing considering that he was having them every day. He's starting to get a little bit of his independence around the house back. And I feel that his spirits are being lifted. He's coming up 1 year post-op and he will be able to start weaning off the immunosuppressant drugs. When the transplant occurred, he has to be on 2 different immunosuppressant drugs for a year post-op. And they do make him pretty tired and feel pretty run down. And we have to keep him safe, especially because this was all happening just as the pandemic was, ending. So that's been a challenge for him in terms of getting any kind of social life back. But we're looking forward to continuing to see the improvement. And if it wasn't for doctors that are willing to and have the heart to keep researching, to find ways that are less invasive, because that was Austin's, big thing is this is so much less invasive than having to go in and have part of, your brain cut out. And as I couldn't imagine as a patient, how scary that would be. So that's Austin's.
So that gets me every time. So that was the first patient and his mom, and these are his seizure counts, the actual data behind the story, right? And that was at the 1 year mark, and now he's almost to 4 years, and hasn't had a, so you can see occasional seizures that are mild, but hasn't had a severe loss of consciousness seizure since the first month, which is amazing, right? And I love the story that someone has to be first. We put that on all the t-shirts for all the employees. And he's shy of speaking in public, but he did do a private video for the employees when we celebrated our 10-year anniversary recently. That was special. And then one of the new things that wasn't in the video that we heard through the coordinator is that both he and his mom are both working again. So
Everyone,
so those are the first 3 patients. Those are individual stories, but of course we went on and we treated the rest of the patients that I mentioned. And so these are the 18 patients now that CIRM funded the first 10 on in the first unilateral study, looking at both doses, low and high. Here, the y-axis is a little different. Instead of showing the raw seizure counts, this is showing a % change from their baseline. So the baseline set to 0, and minus 100 is seizure freedom. And so you can see this is the group medians now in each of the dose levels. So in blue is the low dose, red is the high dose. What you see is 90%, almost 90% median reduction in the low dose, and about 58% in the high dose. Now, if you look at the bottom, you can see the patient numbers. And this is, you see that not all of them have completed the full 1 year. That's not because they discontinued. That's just because people have enrolled asynchronously and they haven't gotten to those time points yet. So again, it's preliminary to see which dose is best. These are not statistically significant differences, but the trend right now is that the low dose is performing a bit better, which I'm happy to talk about later as to why that might be, if it's true. And then you can see the immunosuppression taper ends at 1 year, and then the durability after the immunosuppression has come off has been really remarkable. And we continue to study that as these additional patients come off the immunosuppression as to the durability from a single dose. But because of the trend with the low dose, we picked that for the next populations. So this is the low dose now going into that additional population of people that don't have the scar, where again, it's early, just the first 3 patients, but we're seeing encouraging seizure reductions here. And then into the second trial that CIRM is funding, the 17135, where we have the first 4 patients who got the double dose on both sides of the brain, the low dose in each hemisphere. And here what you can see is 3 out of the 4 have become, seizure-free or close to, albeit early, and 1 in the teal that hasn't responded yet. And we don't know why, and we'll see what happens, and hopefully they can respond later. This patient was featured from a Chicago press. He was the first patient, the 219-001, who is seizure-free now for what we've been told 9 months now and counting. And first bilateral patient funded by CIRM. He broke his spine twice from his seizures, unable to drive. He's depressed, stays home, feels he's a burden. Medication's not working. He's not eligible for a lobectomy because you can't take out both hemispheres. You become completely amnestic. So really stuck. And after NRTX, he's been seizure-free so far, fingers crossed. And his quote is, just maybe we can get a cure out of this. So I'll wrap this up now by saying NRTX 1001 has been well tolerated to date at low and high doses. Significant and durable, we've seen significant and durable seizure control across the different patient populations. There haven't been any cognitive impairments. In fact, we've seen some trends to improvement and improved quality of life. The FDA and the EMA regulatory authorities have acknowledged the potential of this approach. And so we've been granted the RMAT, which stands for Regenerative Medicine Advanced Therapies, and the PRIME in Europe, which stands for the similar to Breakthrough, the Priority Medicines designation, which grants us additional advantages and more frequent interactions with the agencies. And in these interactions, we've agreed with these agencies on the next phase of development, which will be a single phase 3 trial. It'll be a randomized, sham-controlled, double-blind study of cell therapy versus a sham procedure. And this will be the ultimate test in a double-blind setting, rigorously, as to whether this is truly working in a larger population of patients. And we aim to start this trial later this year, hopefully by the middle of the year. So I want to thank everybody for listening. This has been a labor of love for a long time, and it's in large part due to support from CIRM. And I want to thank our patients, our collaborators, and of course the amazing employees, CIRM, and all of you. Thank you. I'm happy to take any questions.
This is absolutely incredible work. Congratulations. I wanted to ask, you see that sclerotic change in the hippocampus and mesial temporal sclerosis on MR. Presumably, as it's repairing in some of those patients, that signal goes away. Do you know if that's happened, or and what do you think the basis is for repair of a lesion?
Yeah, so this is a really good question, and I didn't explain it clearly enough, but so the hippocampus becomes degenerative, and there's a lot of cell types that are dying. But what I was focusing on that slide was the dentate gyrus, which becomes distended and then becomes re-thinned again after cells. And we're not transplanting dentate cells, right? But we think it's the reduction in the seizure activity that's allowing those distorted cells to refine their home, either through migration or through the neurogenesis process, because this is actually one area in the brain where there is new neurogenesis happening. And perhaps those cells can find their place without the seizures. But we don't know that. I mean, this is a really good question. And that's a mechanistic area that we continue to study. Yeah.
Thank you.
In the humans, by the way, you don't have that level of resolution on the dentate with MR, unfortunately. So we don't know.
Isabel.
Thank you. Corey, when I was a kid growing up, my mom suffered from epileptic seizures. Most of us didn't know what the heck it was. Very scary. My dad was usually the one that jumped in to save her. Once we were on the road through Los Angeles to Arizona, and she had a seizure. So we pulled up into the USC public hospital, but it was these kinds of experiences as young children that we didn't understand. And obviously my parents didn't that much either. My question to you is super basic. I did not see demographic breakdown on the populations you've served so far, or the groups that you've served so far. Do you have data on those that have gone through these trials? Is it diverse? Do you know that this is a kind of a treatment that works for multiple populations, or do you have to still study that, or where do you stand with that?
Yes, and I didn't present the demographics, and I actually don't recall off the top of my head, but I believe we have at least 2 or 3 out of the 18 who are from the Latino population. I don't remember if they're in California or not, though. But epilepsy itself is an equal opportunist. It affects all populations ethnicities, races, regardless of population. So, but the people that we see are biased because of the healthcare biases that we know we have, right? So these are people that we're seeing that are already at the end of the funnel. They have to be referred, 3 times to get to the place where we're seeing them. So it's biased for people with the health insurance that are not in healthcare deserts. And this is a huge problem the epilepsy community and all, medical communities are facing and trying to fix, but it's a hard problem.
Well, that's critical to know, but when you talk to my community, show me, show me. So I need to see the data.
Yes.
And I need to see the data to know it's happening at my community. And therefore they need to care and they need to engage and they need to push for change. So I really appreciate it. Thank you for that. Bias is something our communities are used to.
Couldn't agree more. And, part of our CIRM applications have the DEI component. So we do spend dollars on DEI experts, and we have a team that's trying to educate, right? With data makes it more compelling, right? So people can start to trust, but getting it out there to communities, especially around these centers, where they can become part of the trial and then spread it to areas that are less, well-served.
Kim, then Mark.
Thank you so much for your presentation, Corey, and for showing so eloquently, both in your words and the words of the mother of the first patient, exactly what CIRM can do for the world, but especially for California. And I just want to nerd out on the scientific question now, which is, if indeed there is a difference between the low and high dose, what are some of your hypotheses about why the high dose appears to be less effective?
Yeah, I don't want to overinterpret it because it's still a small number. But one of the things we saw in animals is once you saturate the brain tissue with cells, if you try to inject more cells into the brain, they die. And the other thing is to get to the high dose, we put in more volume. And the brain is not like the blood, it's very contained and there's not a lot of space. And so you have more pressure and you have more debris from the cell loss by trying to inject more cells, which could potentially, be detrimental to engraftment as a hypothesis, but we don't know that that's happening.
Yeah, could you, excuse me, comment more about the Alzheimer's indication that you're potentially looking at?
Yeah, so it's not a plaque and tangle clearing approach, obviously, but it's known that a subset of Alzheimer's, anywhere from 30 to 50% of Alzheimer's patients have epileptic-like discharges in the temporal lobe, in the memory center of the brain, and they don't know they're having them because it's not frank seizures. The only way you would know is if you have a sleep study with a sleep EEG to detect this discharge activity. And what we know is that the people that have those that have been followed, they have a faster cognitive decline due to what has been hypothesized is electrical activity that spreads the plaques and the tangles. And so we think that by taking the same approach with the same product in the same part of the brain, we can reduce that activity and indirectly reduce the spread of the pathology and maybe slow cognitive decline. The other finding is that interneurons are one of the first populations of cells to be lost in the brains of patients. And the Broad and the Allen have shown this with global transcriptomics, showing that that's very well linked to cognitive decline. And so it's actually it's a subpopulation of interneuron that's the exact same one in NRTX 1001. So there's a few angles of evidence that point to this as an interesting hypothesis to test, which is actually very tractable because we would be putting the same product in the same part of the brain. So we're thinking about this in the years to come.
Because our next speaker has a absolute hard stop, I'm going to ask the remaining people on the board who want to discuss with Corey to carry that on over the lunch break, if that's okay. And I apologize, Corey.
Thanks so much.
I know you also have an
No,
appointment. Yes.
appreciate it. Yeah, thank you for listening.
Thank you. Thank you very much. And he will be followed by Crystal?
No, wait, we're going to take a break now to allow the members in the room to go get their lunch and bring it back to the dais so that in 15 minutes we can get started with the next presentation.
Right. Great.
But I am chuckling after our conversation.
It's sitting.
I got assigned to the.
It works, it just doesn't turn on. Mhm, that's what I meant, sorry. Here.
I'm testing it, I got it.
All right, we're about to resume. Yeah, I just wanted to.
Okay, everyone, welcome back. Enjoy your lunch. We're going to continue with the presentations. Dr. Crystal Mackel at the from Stanford is at the podium and is ready to lead us into the second part. Thank you.
Thank you. Thank you, Vito, and other members of the board. Thank you to members of CIRM leadership and to the public who are listening. It is my pleasure to have the opportunity to share what we think are exciting and impactful results directly as a result of CIRM funding for our work in targeting a deadly pediatric brain tumor, a deadly brain tumor that occurs in children and adults, but tends to afflict children in an outsized way. So this tumor is called a diffuse midline glioma, and it can be characterized as a universally lethal cancer that even in 2026 has no upfront standard therapy options. It occurs in the midline of the brainstem or the spinal cord, also sometimes in the supratentorial region. But it has a predilection for school-age children, but we also see it in young adults, and it also does occur in older adults. Now, we all know that gliomas, high-grade gliomas, all of these are high-grade, are dreadful diseases. Glioblastoma, of course, one of the major challenges in oncology. This is the worst of the worst. So among gliomas, the outcomes are even worse than for your typical glioblastoma multiforme. There are many trials. You can see on the right there, the median overall survival is about 9 months. And at the end of 2 years, there are very few, if any, survivors. It afflicts about 1,000 patients per year in the US. So if you extrapolate that to California, we're talking about 120 patients or so a year in California. It is the most common cause of death in children due to brain tumors. And the only treatment, patient gets diagnosed today, the oncologist who makes the diagnosis goes in and says, you know, I'm sorry, the only treatment we have is radiotherapy. It's palliative. It will extend survival by a few months, but essentially all patients with this disease die. So it's really pretty devastating. Now, the molecular biology, what drives this cancer, has been understood pretty well for over a decade now. It's a mutation in this part of the epigenetic apparatus called histones. And this mutation leads to dysregulation of all the genes that get expressed. Michelle Mange is a neuro-oncologist at Stanford who's been studying this disease mainly by getting tissue from early autopsies of children who die of the disease. And she made a discovery from a very simple experiment and found out that this oncogene, this genetic mutation, causes massive overproduction of a fatty sugar called GD2. It's an abbreviation for something called dicyaloganglioside. Now, my lab, it just so happened, had been studying GD2 and targeting it because this target's also found on a pediatric tumor called neuroblastoma. So when Michelle made this discovery, you can see there on the red, the big expression of the GD2, she knocked on my door and said, aren't you working on this target, developing immunotherapies? And in fact, my lab had been developing a chimeric antigen receptor specifically to target GD2 in that pediatric disease. She said, look at this. This is massively overproduced by this diffuse midline glioma. And I knew about this tumor. Of course, we all do in pediatric oncology. And you just stood there and shook your head because it was hiding in plain sight. We just didn't know enough about the tumor. I said, sure, we have a CAR, a chimeric antigen receptor that we've been working on, and we'd be happy to collaborate with you to see whether we can develop this for this deadly brain tumor. Now, just a little primer on CAR T cells. You guys, as members of the CIRM board and the interested public, I'm sure you've heard about CAR Ts. But for the most part, these are intravenous autologous therapies. And they start when you get a lymphocyte from a patient with the disease that you're going to treat. And most of the time, that's a cancer patient. So you take the lymphocytes, usually from the arm of these patients, and you run them over to the lab, and you do a genetic engineering procedure. This basically means you take the nucleic acid for that CAR receptor, and you insert it into the genome of those lymphocytes. You expand the lymphocytes, millions of cells, and then you go back to the clinic, and you infuse the cells. And most of the time, the cells come out of the vein, and they go back in the vein. Now, this work on CAR T cells has been really very active over the last decade. And we now have 5 different CAR T cell products approved for the treatment of cancer. But they're all approved for liquid cancers, leukemias, and lymphomas. And they've really transformed the treatment landscape. Now, people like myself and Dr. Okada and other investigators, we want to figure out, can we use these therapeutics that have been so powerful for solid tumors because our laboratory studies say that we can. But thus far, the proof of concept has not been demonstrated in humans. Now here is what we call an organoid. It's a 3D structure of a GD2 positive brain tumor, like DMGs. And the green blobs there are live tumor cells. The red are dead cells. And the blue are CAR T cells, taken from the same patient that the brain tumors come from. And you see those little blue cells moving around, moving from cell to cell. Those are killing the tumor cells. And this is happening over the course of about 24 hours in the test tube. And this is the power of CAR T cell therapy, because you basically are using the Mother Nature's immune system that can induce this recursive killing. The question is, we know this can happen in the test tube. How do we deliver this to patients? I told you that intravenous delivery is the typical approach, but these are brain tumors, and we want to get the cells to where the action is. And so investigators at City of Hope had demonstrated that you could put CAR T cells into the CSF and that they also perform pretty well there. And in our own studies, they're more potent if you put them directly into the CSF rather than into the vein. So at this point, we knew we had something that could be powerful, but how do we actually translate it? So this is where our CIRM funding came in. We received the CLIN2 in 2020, 2021, and we opened a CIRM-funded phase 1 trial of GD2 CAR T cells. And of course, it was the first in human, first in child, to assess feasibility and safety, but we really were hoping also that we'd see activity. Now, this is a complicated schema there. You can see it. The patients have to have that mutation because that tells you they massively produce that target. And we're manufacturing these cells the way CAR T cells are typically manufactured. And we decided to hedge our bets. We said, let's give the first infusion intravenously, because that's the way we studied it in the mice for the most part. But if patients needed a second infusion, we could give that directly into the brain. And so we had the Ommaya catheter placed that just sits under the skin and can be accessible. And this is all work done with a large team at Stanford. Now I'm showing you some of the remarkable clinical responses. This is a young man who you can see the tumor had impacted his ability to smile. We gave him a dose of the intracerebral CAR T cells. His face now starts to work. Here he is suffering from basically what we call paresis or weakness and spasticity of the right side of his body because of the involvement of the tumor. He's treated now with a dose of those CAR T cells given into his brain. 2 weeks after this, you see him in clinic with an almost normal gait. This day, he goes for a hike with his sister. Here's another patient, a 17-year-old who came to us with the Pontine version. And you can see at the red arrow there, this great big ugly tumor sitting in the middle of his brainstem. We observed very significant reduction, so much so that by 8 months, we saw no evidence of tumor. That kind of funny shaped blob in the middle, that's a normal ventricle. And so we're left only with a little scar in his brainstem. More important than the radiographs or the MRI is his clinical status dramatically changed. When he came to us, he could walk, but he was in a wheelchair if he was going to go any distance. He had to, stay home from high school. And upon treatment, he gradually recovered his ability to walk and do his normal activities. Here he is 1 month after the first infusion. Can you guys make that go? Video? Okay, there you go. See, his dad, has to help him that's 1 month after his first infusion. You can see 8 months, he's walking normally. He's now, graduated college, traveled Europe, has a forestry degree and is doing remarkably well 5 years out. So here are the clinical results from this first arm where we gave IV infusion followed by infusion directly into the brain. And what you're seeing there is the degree of either growth or shrinkage, maximum shrinkage or growth after the treatment. And, given that this is a tumor for which there are no therapies previously reported that can cause any shrinkage, the fact that we saw 4 patients on this arm with more than 50% tumor shrinkage was really quite paradigm shifting. And some of the patients with less shrinkage also experienced neurologic improvement. Now, if you remember the 9-month median overall survival, here on this arm, we saw median overall survival of 20.6 months. And as I mentioned, this was associated with really significant clinical benefit and this favorable survival. So that's the good news. The bad news is, like it is so often in cancer medicine, your active therapies also often bring along toxicity. And you're always trying to thread that needle between activity and toxicity. And so patients on this arm A also experienced some significant toxicity. Most notably, on the left, what you see is something called cytokine release syndrome. And this occurred after the first cycle where we gave the intravenous infusion. And some of this cytokine release syndrome was grade 3 and grade 4, and very difficult to manage in a patient with brain tumors. And so we knew that this was a problem and could not be tolerated. We also determined that there was other toxicity related to tumor swelling, shown on the right, which occurred whether we gave it IV or ICV, intracranial, and we learned how to manage that. I also want to highlight the lymphodepletion there. So CAR T-cell therapy thus far, or any T-cell therapy, has not worked unless you prepare the host by reducing their natural immunity. You're essentially making space for the new T cells to give them an advantage. We call that lymphodepletion, and it's typically chemotherapy. So on Arm A, we gave 1 dose of lymphodepletion before the IV dose, and then we didn't give it anymore. So why did some patients do better than others? So this is where the CIRM funding is so critical. Of course, it allows us to actually conduct the trial, but for these early phase trials, if you just conduct a trial and you report outcomes without understanding the why, you have really failed, because the whole purpose is to understand how these therapies fail, what are the mechanisms of resistance, why do some patients do better than others. It's the whole process of iterative improvement. And so we work very hard to understand all of this, and we study every patient in great depth. And what we learned from this Arm A was that the patients who did the best were the ones whose CAR T cells lasted the longest. So what I'm showing you there on the left is patient number 10. This is the patient who had the durable complete response. And in the blue are his CAR T cells circulating in his bloodstream at multiple days after the first dose. And you can see he had these cells detected for about a year. The red squares are the level of tumor DNA in his CNS. And you can see his tumor DNA went away and it stayed away. Look on patient on the right, that's patient number 6. This young woman had a very impressive response also, but it disappeared over time. And around 9 months, she experienced a relapse. And we noticed that her relapse, you can see the growth of the tumor as the red squares increase, coincided with the loss of the CAR. And so it was a relatively simple hypothesis that these things need to stick around for a long time in order to work. So on Arm A, 13 patients treated, 1 dose of lymphodepletion, IV infusion, and then subsequent intracerebral. We had that favorable overall survival, but we had a problem with toxicity, and we had the correlates that suggested we needed persistence. So we had to amend the trial because we had to deal with the toxicity. So we opened Arm B, and at the time, our mouse studies had demonstrated, as I mentioned, that it's more potent if you give the cells directly into the brain. So we said, why don't we just bag the IV dose? And why don't we just administer the cells into the brain? And the feeling was you don't need lymphodepletion because that's really to open the space in the systemic system. We didn't feel we needed to open the space in the brain. So we gave no lymphodepletion and we just gave these cells once a month into the brain. Well, we saw responses, but they were not sustained. In fact, the overall survival was 14.8 months, and it was really heartbreaking because patients would recover function, and then about 3 months in, they'd go back to where they started. We'd gotten rid of the toxicity. We didn't have any cytokine release syndrome, but we had lost our efficacy. And when we went back and said, are the CAR T cells persisting? The answer was no. Here is comparing the Arm A patients looking in their bloodstream at the CAR T cells. You can see the good levels that these patients have for many months after the first CAR T cell infusion. Look at Arm B. We got a little blip in the bloodstream, and then they were all very, very low. So we're immunologists after all, and we said, what is happening? Are our patients rejecting our CAR T cells? That could explain what's happening. So we went looking for immune rejection. And here we're looking for the presence of antibodies against the CAR T cells. And we can do this in the laboratory. We just take a T cell, we express the CAR on it. Then we put the patient's serum into the test tube and we look for binding to the CAR T cells. And what we saw was that most of the patients on Arm A developed these antibodies, and they developed them at the time point at which they lost their CAR T cell. Let me show you an example here. Here's patient number 9. At the top there is green means she was experiencing clinical improvement, and red means clinical progression. There's your CAR T cells in the bloodstream. You can see they're at high levels and they last for about 200 days or so. And then you lose them. And there are the red squares showing how the tumor recurs. Now on the bottom, the blue isn't CAR T cells. On the bottom, the blue is CAR T cells, but the red is the antibodies. And so you see these antibodies come up right at the time we're losing the CAR T cells. And so we now really had a very firm hypothesis that immune responses, immune rejection of the CAR T cells, are limiting our CAR persistence and preventing disease control. That means that the lymphodepletion was important in Arm A, not just to provide space for our intravenous infusion, but to prevent rejection. And so you can imagine now, what did we do on Arm C? We gave intracerebroventricular CARs with lymphodepletion. And we've been very excited to see the results on Arm C because our median overall survival has not been reached. This means that we are seeing median overall survivals that are really, I would argue, could be paradigm shifting if they are to persist. And we've got very significant follow-up at this time. We don't have toxicity. It's non-dose limiting. And when we look at the anti-CAR antibodies, we still find them, but they're at lower levels. And so we're at the point of tweaking. We're tweaking it to get ready for our pivotal trial. We've just opened an Arm D that gives us a little more consistency in dosing, where we've added rituximab to get rid of the antibodies altogether. And we think now that this is the regimen that we would ultimately want to test in a pivotal trial. So the current status, this funding has absolutely allowed us to perform this potentially paradigm-shifting trial in diffuse midline glioma. It demonstrated safety and significant clinical activity of these CAR T cells for this universally lethal brain tumor for which no standard upfront therapies are available besides palliative radiation. And our patients appear to be living longer and experiencing better quality of life following the treatment, which is exactly what we all want. We've learned that the target product profile requires sequential intracerebral infusions and sequential lymphodepleting therapy we believe, along with rituximab. We'll see for sure. Based upon these promising results, the therapy has been designated in a regenerative medicine advanced therapeutic by the US FDA. And because we have now that access with the FDA, we've completed a comprehensive, what we call RMAT Type B meeting with the FDA, which is basically the meeting that discusses plans for a pivotal trial. And that meeting was very informative and also gave us a lot of confidence that the regulatory risk of this product is low. The FDA was very favorable, gave us clear guidance on our CMC that we need to meet, all of which are quite feasible, and clear guidance on what the clinical trial will look like, a single-arm clinical trial with overall survival as the primary endpoint and neurologic improvement as a potential key secondary. So what are the next steps? Well, we're going to complete this phase 1 trial at Stanford, finish Arm D, and confirm that that is the TPP that will move forward. We need to move from a single institution phase 1 to a multi-site phase 1. We don't need to do a massive phase 1. The phase 1 at Stanford has been almost 50 patients so far. As we move to the multi-site, we need to train investigators how to administer this therapy and ensure that we can deliver it safely. And so the first step would be to conduct a small multi-site phase 1 to ensure reproducibility of the results. At that time, that would be done by a centralized CDMO, and again, train our non-Stanford investigators. All of this is in preparation for launching a multi-site, potentially pivotal single-arm phase 2 to demonstrate efficacy, and ultimately with the goal of filing a BLA. We believe that this can be completed within the finite timeline that CIRM is under. This success has emphasized, however, a critical need to bring active cell and gene therapies to market for rare disease, and CIRM knows very well that that is a challenge. This is not as rare as SCID or some of these other diseases, but it's still a rare disease in the eyes of biopharma. And so one of the things that we've been doing is thinking hard about more innovative economic models so we can get it over the finish line. This is a paper we published a couple years ago. You'll see some familiar names on there. Here are a bunch of scientists sitting down, taking time to talk about new economic models. This is such a strange time to be doing what I do because in all my career, it's always been the science that has held us back. And what we're seeing now is that the scientific breakthroughs have delivered. It's the economics that's holding us back. And so we've got to do the work to figure out how to get this over the finish line. Toward that end, let's see, I'm getting another little, can you advance for me, somebody? We've launched a nonprofit, Access for Kids, with a nested public benefit designed to bridge this pharma valley of death. And we hope that this, we're in the process of licensing this asset from Stanford into Access for Kids. We think that this will give us a new model to be able to deliver this at greatly reduced cost. Contributors, there's so many people at Stanford. Michelle Mange and myself have been working hand in hand, but with so many other investigators from the Pediatric Cancer Cell Therapy Group, the Neuro-Oncology Group, the neurosurgeons, the correlative studies, and the regulatory team and other collaborators. But we could not do this without CIRM funding. It's been absolutely essential, and we're incredibly thankful for all of what CIRM is doing. Stop there.
Anne Marie, you have a comment?
Thank you, Christelle. Remarkable breakthrough, remarkable way of thinking forward about affordability and accessibility via multi-center trials. Do you think this breakthrough can also apply to other tumor in children that are similar and that have a similar poor prognosis?
Yes, absolutely. In fact, you'll see a paper that just came out about a week ago that I wrote, an editorial about how the reason we're launching that nonprofit because it is our belief
Mhm.
that cell therapies for pediatric cancer is a missing piece to the puzzle. We've had a major market failure for children's cancer. We still treat those patients with drugs developed in the 1980s. So this is our first asset that we will develop, but there are other assets in the pipeline that if they were occurring in other diseases, their efficacy is good enough to get over the finish. So I absolutely believe that to be true. And other brain tumors, I want to say. You know, the learnings that we have made by studying this rare pediatric and adult, but mostly pediatric disease, are directly informing our efforts for glioblastoma multiforme. So I do believe there will be learnings that go beyond this disease.
Great. Isabel, please.
I was going to say yay.
Thank you.
But you didn't mention the age of the populations across, and I know you mentioned more as a pediatric disease. Is the efficacy the same for the adult population it is with the children? Is it a little more toxic with children because of being younger, et cetera, et cetera?
Boy, is that a great question. Because if you ask most people, what disease does this, what population does this disease occur in? They're going to tell you the pediatric oncology population. But the World Health Organization in 2016 deemed this a whole different type of brain tumor. And what has happened is that now that we look for that genetic mutation, we find that it actually occurs in more adults than you thought. Now, if you don't have a therapy that matters, you never see those patients. But now that we have this therapy, we are getting calls from patients who are 30, 40, 50, 60. We've modified our pediatric trial to open on the adult side, and we're now treating up to the age of, I think, 55. So this is a funny situation. This is one disease where we have more experience in children than in adults. I've been so used to the other, where children are the afterthought. So we will learn. We are very hopeful with the FDA that we can get an indication for all patients with this rather than limiting it to a certain age. And so it'll be important as we move forward with our trials that we do enroll both children and adults to be able to have that complete data package.
Can I ask a follow that might not be a great scientific question, but is the indicator in adults the same one as it is in the pediatric population, and they just lived with it longer or down the road or et cetera?
Oh, it probably didn't start when they were children. It's just as aggressive in adults.
Okay.
For whatever reason, it occurs in an older population sometimes.
Fantastic. I was just wondering, assuming your CAR T cell is focused on the epitope for that fatty
Yes.
acid, so why is it more effective in this disease versus neuroblastoma that you mentioned that also produces?
Yeah, actually, there's been some exciting work with GD2 CAR in neuroblastoma. Most of it's come out of Europe from the group at Rome. This is moving forward in Europe for neuroblastoma. I think we will see an approval. We've decided to focus our effort on diffuse midline glioma, but it is proving active in neuroblastoma. Neuroblastoma is a more complicated and crowded space because there are many other therapeutics out there, whereas for this disease, there are nothing else. So it's from a clinical development path, it might be a little more straightforward, but it has activity in neuroblastoma. All right. Thank you all for your time.
Crystal, thank you. Okay. We're going to move on. There's not going to be a chair's report. The board met only a month ago. It's
Changing the order.
okay. I was just going to pass over this agenda. Give me a second, Scott. Because it's only been a month since the last board meeting at a time of holidays when not much happened. There was the nucleus forum, but our president and CEO is going to report on that. So we'll skip over the chair's report. Now, Scott.
Sure. We're now going to actually return back to item 6, the discussion of the process for preference setting, to resume any continued board discussion and then take public comment as well. So I know that we still have Rosa on the line. We have JT here at the mic ready as well. So I guess the first would be to, if the board has any continued public,
any residual or de novo questions arise from any of the board members? related to Rosa's presentation on preferences. Okay, so seeing none,
All right, we will now,
you can ask if there's any, oh, Lana, right? If there's anyone in the public.
all right, we will now open the floor to public comment. If you're joining us in person, please approach the podium, state your name for the record when ready, and keep your remarks within 2 minutes. For the public, for the participants on the phone, press *9 to raise your hand. When called on, press *6 to unmute yourself. Please state your name for the record, and we'll keep your comments within 2 minutes as well. Is there anybody in the public that would like to make a comment? All righty, we're gonna go to the phone. The phone number that ends in 6596. Would you please unmute yourself? You will have 2 minutes.
All right. Good afternoon. My name is Yvonne Chen. I'm a professor of immunology at UCLA. I'd like to thank you for this opportunity to speak. I'm very grateful that the board has already read the public comments that were submitted prior to the meeting, so I will not repeat those points here. I just want to underscore that the main issue really is not just a lack of clarity in communication, but the actual consequences of PDES and CLIN2 preference system. The list of funded grants show a complete absence of well-proven modalities, such as CAR T-cell therapies, whose potential Dr. Makel just beautifully illustrated for us. The list of funded grants also shows a complete absence of therapies targeting cancer altogether. I think this clearly demonstrates the exclusionary nature of the system. As an example, I submitted a PDEF application for a CAR T-cell therapy for glioblastoma. That's a CNS disease that's currently incurable. By Rosa's rubric, this grant should have received a score of 4 because it also completed a pre-IND meeting, and yet it has been triaged twice in the PDEF system without review. I think it's important to note that even though the preference system is not designed to exclude, the effective consequence is that it now excludes potentially life-saving modalities for high unmet medical needs from having a chance to reach patients. I thank you very much for this opportunity to share my opinion, and I thank you again for your time in guiding CIRM's trajectory.
Thank you, Dr. Chen.
Thank you very much. All right, the phone number that ends in 2019. Would you please unmute yourself?
Good afternoon. My name is Dr. Theodore Nowicki. I'm a California resident and a pediatric oncologist at UCLA, and I'm also a physician scientist who treats children and young adults with aggressive sarcomas using cell and gene therapies, many of which have been made possible through CIRM support. I thank everyone for the opportunity to speak today. I'd just like to reiterate a point that I made in my submitted comments, which is that with the patients that I cared for, I recently treated a teenager with relapsed sarcoma whose family simply asked the question, is there anything left? Is there anything left for treatment? And the only honest answer that we could give was the types of cell therapies that have been made possible by years of CIRM-funded academic work. And I just wanted to reiterate that point that my colleague, Dr. Chen, made, which is that under the current scoring guidelines, it becomes, highly mathematically unfeasible to have any cell therapy for cancer funded under the current scoring rubric. And I just wanted to offer 3 observations regarding the current rubric. First, the current preference-based selection appears, as was mentioned before, intentionally or otherwise, to exclude these kinds of proven track record cell and gene therapies. And I know the point was made about, patient outcomes. These are treatments that have been reaching patients and continue to reach patients for all of those of us that have been involved in this. Second of all, the practical reality that deserves consideration is that academic cell therapy programs that have been meeting milestones often don't really have any viable alternative funding pathways outside of CIRM to advance these into clinical practice with our patients. And this is, especially poignant in the current national funding environment, which has become much more constrained and fraught with lack of stability. And finally, the other issue is that within the CLIN2, there have been a lot of the weighting has been shifted, the funding towards private entities rather than towards.
Thank you so much. You're up. And then the phone number 6063, if you would unmute yourself. You have 2 minutes. (6063), if you would press *6.
Yeah, hi, can you hear me?
I can, go ahead.
Okay, thank you. So my name is Anthony Rivas. I'm a professor of medicine at UCLA also, and I want to thank the CIRM board for the opportunity to talk on the CIRM leadership and Dr. Canet Aviles for the discussion today. Addressing to you, I sent written report, written script, but it's about the CLIN2 that we have submitted twice with 82 pages. We knew about the priorities, but diseases, they called for full applications, and it's not been reviewed scientifically twice. It's for a gel to regenerate the skin stem cells for wound healing, which has an incidence of 1% of Californians, more common in marginalized populations, but has limited interest of commercial development. There's no existing drug therapy for this condition, and we designed a clinical trial that would go from phase 1 to phase 2 randomized, and to move the therapy forward and bring it into California as soon as possible. We also addressed the affordability and commercialization request. We work with colleagues at the UCLA Anderson School of Management for this. None of this was scientifically reviewed by CIRM. If the goal of CIRM is to develop regenerative medicines for diseases that affect Californians, why would a grant like ours only check 2 preferences, 1 being in California and 2 having a prior CIRM grant? Thank you.
Okay, thank you so very much for your comment. All right, the phone number 6583, if you would please unmute yourself.
Good afternoon. My name is Christine Brown. I'm a professor at City of Hope, and I lead a translational research program focused on clinically advancing CAR T-cell therapies for incurable brain tumors. And I think it's very timely after Dr. Makel's presentation today. I wanted to make and share sort of 3 points based on recent experiences. First, I'd like to comment on the new pre-submission review process. I recently submitted a DISC-4 pre-application that strongly aligned with all published CIRM preference topics, including neurological diseases. The project proposed a cross-disciplinary systems biology approach to understand the complex biology of brain tumors by interrogating unique patient samples from multiple brain tumor CAR T-cell clinical trials, including those developed and executed with CIRM funding. Despite this alignment, our pre-submission application was administratively triaged without evaluation comments. This raises questions as to the administrative criteria used without independent scientific review to select only 17% of the submitted applications. And, I and our colleagues would advocate for greater transparency around the administrative triage criteria, allowing teams to better align priorities with CIRM goals. Second, I'd like to comment really on the important clinical impact CIRM has achieved. Our success in advancing CAR T-cells for incurable brain tumors, as you also heard from Crystal Makel, would not be possible without CIRM's partnership. Our project at City of Hope was the first CAR T-cell therapy supported by CIRM. We treated our first patient within 3 years of a Tran-1 award. We demonstrated that CAR T-cells can mediate a complete response against recurrent glioblastoma, and we have patients living years.
Okay, thank you for your comment. I don't see any other hands raised.
Okay. Yes, Isabel, you want to add something? Oh, no, you're still on.
No,
then.
I was in pain for her.
Okay. Okay. So I want to thank all the members of the public who spoke and especially the authors of the letters. You can tell from the testimony here that the board members have read your letters. They were carefully written. They were very informative, and I hope you are left with the impression that your comments will be seriously considered in the next couple of months. Did you want to say something, sir?
Excuse me, yes,
As part of public comment?
Yeah.
Yeah.
On behalf of my colleagues, it's not that we are trying to skew all the grants towards cancer research.
Start by identifying yourself for the
Right,
record.
excuse me, I'm Hideo Okada, professor at UCSF. Thank you.
Well,
So one of those I provided letters. So it's not that we are trying to skew all the grants going to the field of cancer. All this is important. There are many ways to help people. I think my clear point is that implementation of those new priorities all of a sudden in a preclinical and clinical stage, that is, in my opinion, is the issue because every project has a developmental stage. And then as clearly Dr. McCall presented, the autologous cell therapy is still a platform. For important thing is for us to demonstrate reproducible safety and efficacy using the reliable platform. But clinical trials has incremental nature that we ask one new question each time. So we have to rely on reliable existing platform to ask one important novel question. That's how clinical trial moves. So therefore, if you implement in vivo gene therapy, non-viral iPSC, all of a sudden in preclinical PDEV and CLIN2, that can be rather a bit disruptive rather than constructive. That is my question, and I really appreciate if the board can consider this aspect in potential revision of the priorities.
Thank you very much, Doctor. I appreciate your comment. Okay, I think we have reached the end of this agenda item then, correct? Good, okay.
12.
Yes. Oh. We're moving to agenda item number 12. Okay, so I'm going to introduce our Associate Vice President for Preclinical Development, Dr. Shyam Patel, to come to the podium. This is going to be an exciting presentation. I hope you all find so, on our rare disease platform proposal.
Thank you, Dr. Mbasiani. And while I'm waiting for the slides to come up, so I'm Shyam Patel, and thank you to the ICOC for this opportunity to present the rapid funding opportunity to you today. And thank you to the Science Subcommittee for your endorsement a couple weeks ago. I'm presenting this funding opportunity concept on behalf of my colleagues, Dr. Jim Campanelli, who's in the audience today, and Dr. Lisa McGinley, who's on Zoom. And the rapid funding opportunity stands for Rare Disease Acceleration through Platform Innovation and Delivery. Full disclosure, I will be making a reference to cars, but they are of the boring driving variety. And advanced apology to the Science Subcommittee to have to hear that analogy yet again. Okay, so this presentation is going to focus quite a bit on the background and motivation for this funding concept. And then I will go into the structure and scope of the program, ending with the timeline. So the rapid funding opportunity is responsive to the SAF Goal 3, which was to advance 4 to 7 rare disease projects to BLA. In that goal, we had 2 recommendations, and this is for the second recommendation, which was to advance a new model for rare disease therapy development that is based on platforms. And I will say platforms and rapid 100 times, and we'll get through this one step at a time. Okay, so as you all know, there are over 10,000 unique rare diseases that affect the world population today. While each rare disease affects a small group of people, collectively, over 30 million Americans are living with rare disease today. Over 50% of those rare diseases manifest themselves in childhood, and 80% of them have a genetic basis. Now, the vast majority of cell and gene therapies that have been approved and are in the market today are for rare diseases, but 95% of rare diseases have no approved therapy. Why is that? And we all know that. It's because it's, the drug development process, particularly for advanced modalities, is time, resource, and cost intensive. And it makes it very difficult to be able to develop therapies when you're looking at thousands of diseases. So this gets us to the point of, this is reinforced here again in this slide, which is that even for gene therapies where the development pathway is a little bit accelerated compared to small molecules and biologics, it could still take 5 to 10 years to get these therapies through the development life cycle, from candidate discovery through preclinical development to clinical trials and FDA approval. And as I mentioned, because you're largely repeating the testing process wholesale for each candidate, it's very difficult to develop these therapies in any parallel format. And you often see these types of staggered or serial development of gene therapies across academia and industry, because you just cannot commit the amount of time, resources, and funding to be able to do it in parallel. What if you could speed up the development of a genetic therapy? We saw an example of this, and would this actually result in a scalable solution? So we saw an example of this recently where the Children's Hospital of Philadelphia team developed a base editing genetic therapy for baby KJ within 6 months of diagnosis. While that was a very remarkable achievement because they had taken a novel gene editing technology and drove it to the clinic in 6 months, if you look at their testing timeline, they had to do the large majority of the testing that you normally would do for a gene therapy. This includes testing in, sorry, is the mic still on? Okay, thank you. They had to do all the testing in small and large animal models and all the manufacturing for this therapy. And because it was an N of 1 therapy for a single patient under a single administration, you would have to repeat all of that testing again for the next baby KJ. So while it was sped up in time, it was not efficient in the use of resources and dollars. So this is where I'm going to pause to talk a little bit about cars. And so imagine in your mind 2 cars. One is, I'm not sure I can do this with a straight face. So one is a Honda CR-V, which is an SUV that I drive. And the other is a Honda Civic, a compact sedan. These look like 2 completely different cars. They have different functions. They're meant for different lifestyles, but they're in fact based on each other. The Honda CR-V was designed and built on the chassis of the Honda Civic. And that was a deliberate strategy where every single generation of the CR-V is built on the same generation of the Civic. That's a deliberate strategy to apply platform efficiencies to every single stage of the product development and product life cycle, starting with design, testing, getting into manufacturing, and maintenance of those vehicles for every generation of those cars. What would happen if we took that sort of an approach, where you're realizing platform efficiencies is every single stage you see here, from discovery through preclinical testing to clinical testing to approval? What would that look like? And so that is the concept of platform-based therapies, where you'd have multiple related therapies for multiple indications that are rapidly advanced to patients by leveraging common components, technologies, data, and resources. This is done to a certain extent today, but what I'm going to describe in the next couple of slides is how to do that efficiently across the entire life cycle. So here are a few illustrative examples. They're not meant to be limiting, but to just give you an idea of potentially platformizable genetic therapies. There's a non-viral gene editing, RNA-based therapies, and AAV gene delivery. We're going to key in on the gene editing. I'm going to carry forward that baby KJ example a couple more times here. So if we look at CRISPR gene editing, there are 3 basic components to that type of a therapy. There is a delivery vehicle. It could be a non-viral lipid nanoparticle, an editor enzyme, which does the job of modifying the DNA, and as well as a guide RNA. The guide RNA's function is to guide the enzyme to the appropriate sequence in the gene. by varying just that guide RNA, you can actually create hundreds of different therapies that target different mutations. And so with that, I'm going to walk through what is an ideal state of platform-based therapy development and approval, and where we're at the moment. So as I just described, it's relatively straightforward to use a platform like CRISPR to discover a large number of candidates. So let's go back to the baby KJ example. Baby KJ has a urea cycle disorder where it had a mutation in one gene. There are 6 other known genes where a mutation causes a disruption in the urea cycle process. What if, instead of developing a therapy one at a time, you developed a therapy for all 7 of those genetic mutations at the same time? So effectively, you're looking at 7 different candidates, or more actually, for 7 related indications and 7 patient populations. Okay, so in this simplified example, let's just assume that by varying the guide RNA, you could create a base editing therapy for each of those 7 mutations. Please pardon the fact that I stopped at 3 graphics here. And then, so you can develop those therapies relatively, you can discover those therapies relatively quickly. Where it gets challenging is when you get to the preclinical testing phase. So here, what you would do in this ideal state is reserve the bulk of your preclinical testing, your animal testing, your in vitro assays, a lot of your manufacturing feasibility to one candidate. And then you would do the necessary in vitro testing for the rest of those candidates. So here, you're basically limiting and using the data from one candidate to justify the platform, but you're supplementing that with data from additional candidates that's maybe only in vitro testing. Then when it gets to the IND and clinical trial stage, instead of submitting a single IND for each of those candidates to do a single candidate trial, the FDA will allow you to submit a master clinical protocol driven by a master clinical IND, which will allow you to do an efficient umbrella trial design where you can test all these candidates at the same time in an umbrella trial. And you can also add additional candidates as new mutations arise in those patient populations. Finally, instead of submitting a BLA for every single candidate, the FDA would allow you, FDA would approve the platform. So they're approving the platform, which would allow for additional candidates to rapidly be developed and approved. So that would be the ideal state of applying platform-based approaches across the entire life cycle of drug development if you were to create Hondas and CRVs. So with that in mind, the prevailing thought so far has been to get to this sort of an ideal state would require a lot of iterative steps where you realize platform efficiencies at one step in the process and you stack all those efficiencies to get where you need to be. For example, right now I'm working on managing an award that is a gene therapy. And that company actually has another gene therapy that has a similar structural elements that's already in the clinic. And they're able to leverage some of the data from that clinical candidate to reduce some of the animal testing for their existing candidate. However, to realize this sort of a strategy would require a deliberate approach. So I mentioned the CHOP team before and the urea cycle disorder in the baby KJ example. The CHOP team has in fact executing on this strategy. They have gone to the FDA in a pre-IND meeting proposing this exact approach, which is parallel development of genetic therapies for all 7 urea cycle genes to do a master protocol clinical trial, and by reserving the testing, the bulk of the testing on a subset of those candidates. They have pre-IND buy-in. The pre-IND meeting minutes have been published and shared with the public. And on top of that, a couple months ago, the FDA, partly inspired by the baby KJ example, put out a very preliminary blueprint that they call the plausible mechanism pathway. The plausible mechanism pathway describes how platform-based genetic therapies for rare diseases can be rapidly developed and approved as a platform, effectively what is described in this slide. And so at this point, while a lot still needs to be done, much still needs to be worked out and proven, there appears to be a positive feedback loop between regulators and therapy developers on how to get to this vision of a platform-based approach for rare disease therapies. So I'm going to take a minute to step back and talk about how we got to this stage. May I please have the slide advanced? Thank you. So the CHOP team's work has been entirely supported by a visionary set of NIH funding opportunities that were issued 3 years ago. So the NIH issued a couple of funding opportunities that asked a very simple question for proposals. Bring us multiple gene editing therapies for multiple diseases, and we will fund you to IND. And so that was their approach. So Baby KJ is actually a stepping stone in that broader CHOP program approach to bring all those gene therapies at the same time to the clinic, and an important validation. On the regulatory side, as many of you know, a couple years ago, the FDA published platform designation draft guidance. This is driven by the 21st Century Cures Act. Unfortunately, that platform designation guidance is not accessible to most cell and gene therapy developers because it requires you to have an approved product in the first place. However, that has spurred a lot of direct interaction between therapy developers, patient advocates, and the FDA on how you can have efficient regulatory pathways for rare disease drug development. And that's what led to the plausible mechanism pathway publication from the FDA. So you can see how all the pieces are fitting together. And the RAPID program at this point in time is, if approved by the board today, would be positioned to advance platform-based therapies from promise to practice by building robust evidence across rare disease and technology. So the goal for this program is twofold, and that's described in this slide. So the first is scientific and regulatory innovation, and second is patient impact. So for that first goal of scientific and regulatory innovation, the idea here is that this program would support multiple approaches for multiple disease areas and build a strong evidence base of safety and efficacy for these platform-based approaches across these different approaches. However, if it's going to do that, it also needs to ensure that it can demonstrate rapid progression of platform-based therapies to patients where there is direct clinical impact. And that's the second goal of patient impact. Ultimately, all this would result in blazing a pathway that others can follow. So with that in mind, the RAPID program has an objective of creating a scalable model to rapidly deliver transformative platform-based genetic therapies to patients with rare disease. I'm going to spend the next few slides talking about the scope and structure of the program as well as the budget ask. And then I'll end with a couple of slides about the life cycle of the award. So as you know, we have a set of funding opportunities that do span preclinical to clinical development. These include our legacy programs like Translational and CLIN1, as well as our newer programs like PDEV and the modified CLIN2. All these programs have supported rare disease therapies and will continue to do so as we go forward. However, these programs are not currently designed to accelerate innovative high-risk platform approaches. So these programs are designed to support the tried and true regulatory pathway of focusing on a single candidate for a single indication, doing all the preclinical testing and manufacturing, and then conducting a single candidate trial under a single IND. Lots of singles there. This should come as no surprise to you, but the RAPID program will do everything in multiples. And so the idea here is that this program would support the development of multiple candidates for multiple related indications in a master protocol trial where the platform efficiencies are realized at both the preclinical, manufacturing, and clinical stages. So in order to support the goals that I mentioned earlier about regulatory and technical innovation and patient impact, we're proposing two types of awards for the RAPID program. The first is the RAPID Validation Award. This is meant to serve that patient impact goal. And the idea here is that there will be a subset of projects that have pre-IND feedback from the FDA on a platform-based approach. And they would be able to apply to CIRM for funding for a validation award which would take them through all the IND enabling studies, the IND clearance, as well as completion of that first in human master protocol trial. So this effectively spans IND enabling stage as well as clinical stage. We could envision some non-limiting examples of types of projects that might come in. This could include non-viral, liver-targeted base editing therapies for metabolic disorders. This is what I've been speaking about with respect to what the CHOP team has been doing. But there's another broad array of metabolic disorders that could be liver-targeted or targeting specific organs. You could also have AAV-based gene delivery for neurodevelopmental diseases where there has been significant clinical progress to date, but can that be platformized? And these awards will achieve the program objective by demonstrating that you can rapidly bring these therapies to patients, as well as building that evidence base of safety and efficacy for these types of approaches. And I'm going to briefly talk about the structure of the awards. So the candidates that would be eligible for a rapid validation award would be in vivo genetic therapies for rare genetic diseases. The applicant would have to have conducted that pre-IND meeting, as I mentioned, and the outcome for these awards would be completion of the clinical trial for at least 3 candidates. And this PA, unlike some of our other programs, will not specify an award cap, and it will not have a minimum co-funding requirement. This is spelled out in the memo, but briefly, what we're intending to do here is to allow for a range of projects where co-funding is not a barrier, but also put the onus on the applicant to propose a well-justified budget that clearly demonstrates time, cost, and resource efficiencies. And like all of our, like our PDEV program, the RAPID program will be limited to California awardees. The second award type is the Rapid Innovation Awards. This speaks to the first goal of regulatory and technical innovation. So the intent here with the Rapid Innovation Awards is to really push the boundaries of what constitutes a platform. So what we expect here is that as novel technologies are developed, they're developed with a platform-based approach in mind, and that they are using that to expand the reach of platform-based genetic therapies to additional types of rare diseases. And so we will support these projects through that early optimization of the candidates through IND clearance. So this is very similar in scope to the PDEV program, but it will not support clinical trials. So we can envision some non-limiting examples could include non-viral gene delivery technologies to the CNS. It could include next-generation gene editing therapies developed with novel in vitro models. And these serve the platform goal by really advancing novel technologies in a platform-based strategy and continuing to add to the evidence base for what would constitute effective platforms. An innovation award would again be for candidates that are in vivo genetic therapies for rare genetic diseases. We would require that the applicants have at a minimum requested an FDA Interact meeting. This is important because you have to have alignment with the FDA for these types of novel approaches. And the outcome for this award would be a master protocol IND cleared for at least 3 candidates. Like the validation award, there would be no award cap specified in the PA and no minimum required co-funding. And like the validation award and our PDEV program, the awardee would have to be a California organization. To support these types of projects, we are requesting that the board authorize a $100 million budget for this program that would be spread across 2 annual funding cycles. And I'll describe how we may come back to the board down the road for approval to reinvest additional funds if they're recovered from rapid projects. Given the dynamic nature of these projects, we would like to request from the board the authorization to have a supplement budget. This would be a program supplement budget that could be deployed by CIRM in a defined request and approval process to help accelerate funded projects that are otherwise moving and executing on their OMs. So to describe what that would look like over a 2-year period. In fiscal year 26-27, CIRM would request that the board authorize $55 million to the RAPID program, $50 million of which would be deployed to fund up to 2 or 3 awards or possibly more. And $5 million would be reserved for the program supplement that could be deployed over the lifetime of the program. In fiscal year 27-28, we would request that the board authorize $45 million that could be deployed to fund additional awards in the RAPID program. CIRM routinely recovers funds from projects, and if enough funds are recovered from RAPID projects themselves, we may in future years ask the board for approval to either replenish the supplement budget and or open up the program for additional RAPID applications. That would be part of our research budget approval process. So next couple slides, I'm going to talk about the life cycle of the award. This is relatively similar to our other programs. So given the nature of this program and the scope of it, we want to ensure that every applicant has had a consultation with the CIRM team before they submit their application. We don't want them to spend a lot of time on the proposal making process without making sure that the program is aligned with the goals. So what we would expect here is that we are going to require that every applicant have a consultation with CIRM before they can submit that application. The application and review process will be similar to our other programs. It'll require submission of a complete application. There is no pre-submission for this program. If necessary, and the volume exceeds the amount of applications the GWG can review, we would do a positive selection mechanism. This is where the grants working group members select applications that they want to forward to scientific review. And like all of our other programs, all of the applications will be scored by the GWG on a 1 to 100 scale. We'll also utilize our existing award management mechanisms to ensure that RAPID projects adhere to the program objective and expected outcome. So we have been deploying a proactive award management strategy across our entire portfolio. This goes beyond quarterly report reviews to have one-on-one discussions with the awardee to make sure that CIRM is included in all FDA meetings and to create a proactive and collaborative environment with the awardee to move that project forward. To ensure that the project is operating on its objective, all awards will use the CIRM performance-driven operational milestone structure. Here, funds are only dispersed if that project meets the operational milestone success criteria defined in the notice of award. This helps us also ensure that the projects are realizing platform efficiencies and they're continuously progressing candidates, the minimum number required for each of those awards. A really critical component of this program is to bolster the knowledge and data sharing requirements because there's 2 goals that we have for this program. One is to ensure that the RAPID awards can leverage the collective experience and knowledge of our vast awardee network. We have a large active award portfolio of preclinical programs as well as clinical programs. And so here, if you have these RAPID awardees who are creating a new pathway here, they can leverage a lot of the experience and knowledge from our existing awardees. So in that instance, what we expect is to create an internal knowledge network where the RAPID awardees and our other awardees are facilitating real-time sharing of study designs, data, resources, and regulatory experience. At the same time, because this program is meant to blaze a new pathway for others to follow, it's important that we require public data sharing. So the PA and the NOA will spell out what can be shared, when it is shared, and how, and to whom. And so on the public data sharing side, the intent here is to build an evidence base and advance best practices in the broader community and to require sharing of study designs, FDA interactions, and applicable data during the life cycle of the award. So shortly after, so I'll give you an example of what this means. So on the awardee knowledge network side, prior to an FDA interaction, the awardee might share its strategy with relevant CIRM awardees who have similar projects who could offer some guidance on here. After the FDA interaction, they would make their regulatory experience and the learnings available to the CIRM awardees to benefit from that. And they would publish redacted FDA meeting minutes for the public so the public can learn from that as well. And lastly, should the board approve this concept today, the CIRM team will embark on early outreach to applicants. And the intent here is to promote the program, to respond to momentum in the field by having a clear conversation about platform approaches, and to facilitate collaborations that will inform the best proposals that we can get for this program. So we plan to do this by having early interactions with prospective applicants, by engaging with experts and patient advocacy organizations, as well as other funding agencies. And lastly, to convene forums or workshops that bring together key stakeholders to talk about and discuss what platform approaches have been tried, what can be leveraged going forward, and what resources are available to them. So if the board approves this concept today, we would aim for an application timeline that would open the applications mid-year. We're looking at a 9-month cycle from application open to award start, which would mean that the first set of rapid awards could be underway in early 2027. So with that, the CIRM team requests ICOC approval of the proposed rapid program concept with an initial allocation of $100 million in the first 2 funding cycles, fiscal year 26-27 and fiscal year 27-28.
So
Thank you.
You jumped the gun, but I so much appreciate it. Thank you. So we have a motion on the floor to accept the recommendation.
And a second by Marv.
It was second? Yes, thank you, Marv. Okay, the floor is open to discussion on the rapid platform initiative to board members. We'll start with the mover.
I just want to compliment Sean and the entire CIRM team for continuation of developing novel approaches that are innovative and accelerative. So this is has an opportunity to be a fantastic program. So thank you.
Thank you, Anne-Marie.
Yeah, thank you. Great sense of innovation. Have you had a chance to discuss this concept with potential awardees, people who would be interested? And can you give us 1 or 2 examples of how it would look like?
So potential awardees, we've had some conversations with some of our awardees who are trying these types of approaches. We do tend to limit discussions with potential applicants when we do that. We've had a lot of conversations with the NIH and ARPA-H who have similar efforts underway. We had ongoing discussions with the CHOP team on what they were doing, and they were very candid with us even before their pre-IND meeting as to what was happening. So all of that has informed our approach here. And so within our own awardee network, we do know that there are at least a few applicants that could be submitting applications that might involve CRISPR gene editing projects as well as AAV projects. Yes.
I wonder if you've thought of ways of publicizing this, because I think this and other things we've talked about today really need to be out to the public so that the public becomes more appreciative of what CIRM is capable of and maybe more capable of in the future.
Yes. So that, as you know, Amy Adams is our Director of Communications, so we're working closely with her on that, as well as we have reached out to several other stakeholders like Alliance for Regenerative Medicine, reaching out to ASTCT and others, where they can also help raise awareness of this program. One of the things that we really want is for this to start a conversation about what sort of platform approaches have actually been tried beyond what we know about, and that would best inform this project. We would also really greatly appreciate our wonderful 35-member board to be able to promote this opportunity as well.
Any other
Over
board
here,
member?
over
Yeah.
here.
I'm sorry, Chad, yes.
So a few questions. Congratulations, it's great. You know me, I'm all chips in, so I'm very much in favor of this, but I have a few questions. The first is, so there are rare disease projects coming in through other mechanisms with other approaches.
Yeah.
How does this program affect those? And if you're going to use a rubric to make decisions, if for whatever reason you have more applications than think you're going to be able to review, what's the rubric going to look like? So is it going to impact the others coming in through other mechanisms at CIRM, and how, and what's the rubric?
Yeah, wonderful questions as always, Pat. And so I'll tackle the first one. So it really depends on what strategy they're taking, right? If they're taking a platform-based strategy, and they think they can develop multiple candidates, this would be the right program for them. You know, if they're taking the more traditional approach, then the other programs would be appropriate for that. The question about the rubric, yeah.
But we have we have part of the decision-making early, you know, before it gets reviewed, is it filling a gap? This is $100 million investment in filling a gap, right?
Yeah.
With really exciting advanced technologies. So is it not going to impact the checkbox that occurs with the other? Because like, well, we have this other program and this is not using the most advanced approaches. So, you know, thumbs down. I'm just suggesting, I don't know if that would happen. I'm just wondering whether it would.
Yeah, you know, it depends on the types of approaches and the maturity of those approaches as well as to what's possible here. So you could have an instance where you have a project here where it's looking at lipid nanoparticle delivery, for example, right? Which you could potentially platformize, right? And that may make sense for the validation project, right? Or if they're developing a single candidate and they can't actually platformize that for whatever reason, they could potentially use that as part of a PDEV award. If you have something that's very novel, like a brand new delivery technology, the innovation approach might be the way to go rather than going for a PDEV award, right? Where you can do it at a bigger scale. To get to your question about triaging and preferences. So for this particular program, if we got more applications than the GWG can review, it would do a positive selection process. So what that means is that all the applications are viewable by a GWG reviewers and they will pick. So that it would not have any checklist criteria. Now, if the board would like for us to revise the PDEV preferences based on what would be expected to come in for the RAPID program, we can definitely have that conversation in March.
Yeah, I mean, I think those criteria are going to be discussed in March. And what you've described is overlapping, but definitely not identical. And so I think there's some consideration. The second question is real-time data sharing, real-time protocol sharing, which is great. How does that affect institutional commercialization?
Yeah, good question. Yes. So, you know, that's a very relevant question in terms if some of these actually have commercial potential, right? And so part of that is going to be what we're thinking about right now is we're actually implementing a version of the knowledge sharing into the PDEV program as well, which you know from last year. And so it's going to be critical to do so in they can redact really proprietary information that doesn't have IP protection would be one way to start that. In the RAPID program, we could potentially even think about, you know, creating a confidential space if needed to be able to share that type of information if it's necessary. But at a minimum, being able to redact important information would be one way to go. So for example, when the CHOP team shared their pre-IND package and the pre-IND meeting minutes, the proprietary elements in there were some of the manufacturing components because they were using a private party for that. And so those are redacted. But the bulk of the information in terms of the questions they were asking, you know, some of the data they were providing on their assays and their own studies was not redacted. And that is still really informative.
Okay, Isabel?
Thank you. Sham, I know you're not going to be able to answer this, pero I'm going to start. asking for cold, hard facts. We talk about reinvesting any monies that comes back to us, right?
Yes.
I would like to know what comes back to us. So I'm going to ask, and I don't know if that's the chair or Mr. President, but I would like a report back to us by March of the amount of money that has come back to CIRM from which programs that are proved to be, I guess, successful in order to give us money back into our treasury. And then what and where have we decided to reinvest that money? So you're saying it's a long shot, we'll get money that will succeed.
So I'm going to make a distinction here. And so there's two ways that we get money back. Multiple ways. Let's focus on there's the revenue sharing component, which is what you're referring to, which would be revenue coming back to CIRM based on revenue sharing provisions in our awards, right? The other part is that through normal award management processes, awards might end early.
Right.
They may get terminated. And so because we commit funds up front, right? We commit, let's say, $10 million up front, but the award ends early or is terminated, and we didn't deploy 5 million of that, right? That 5 million comes back into the research budget to be redeployed. For this program, what we're saying is that we'll only do that in the event that rapid award funds are recovered, and then we can redeploy it.
Okay, thank you for the clarification. I think this is where some of our skepticism, at least for the public and the transparency for the public, has to be the clarity and the clarification about, because I'm only hearing one thing. Take money out, put it out there, pull it back in to reward CIRM for its investment. That's what I hear. What I hope for, but I haven't seen any reports about whether we have been successful at all. I understand what you're talking about on the other, because those I'm sure we've seen any number of times. So I'm still wanting to ask for a report, if someone can determine what that report's going to look like, and hope it's not going to be so difficile. Go back 20 years. How many?
Isabel, we'll be happy to come back and report on that question.
Thank you.
Good. That's a nice answer. I don't see any other hands in the room. Is there any board member on the Zoom that wants to? No. Okay, then I would like to invite members of the public. Can't offer you a platform, but we can offer you a microphone to make public comments. Or on the telephone. And I'm being told there are no hands. Okay. If there are no other comments from board members, we can proceed to a vote on the motion, right, made by Mark Fisher Colbury to accept the RAPID program.
All those in the room in favor say aye.
Aye.
Those opposed say nay. Any abstentions? And I'll poll the members on the Zoom. Judy Chow?
Aye.
Thank you. Deborah Dees?
Yes.
Elena Flowers?
Yes.
Jeff Golden? And Jeff, you may be muted. I'll come back.
Sorry, yes.
Linda Malkus?
Sorry, I'm there. Yes.
Okay, thank you, Jeff. Linda Malkus. Linda, perhaps you're muted as well. I'll come back. Joe Panetta?
Yes.
Thank you. Carol
Linda
Watson?
is yes. Sorry, the laptop was being a pain. Sorry.
No problem. Thank you, Linda. And Carol?
Yes.
Carol Watson?
Yes.
Great. Thank you very much. Thank you, Mr. Chair. The motion carries.
Thank you.
Thank you. Looking now to agenda item number 13. Yes, sir.
Requesting a 5-minute bio break.
Granted.
Thank
Of
you.
course. I mean, I'm a urologist. What can I say? I put my business card in the bathroom, if you're still here, when this.
Much appreciated. If everyone can start taking their seats.
You can hang out there. I don't mind.
All right, we're just about to resume for those on the Zoom. Stand by.
Okay, welcome back from your break. We are now up to agenda item number 13. And to introduce this item, I'm going to have the chairman of our science subcommittee, board member Mark Fisher-Colbury, will have some remarks and make an introduction. Thank you, Mark.
Thank you. I'm pleased to introduce the Director of Grants Management, Doug Kearney, to discuss a minor modification with respect to our awards management policy that covers elements around rates for facilities overhead. So with that, Doug, take it away.
You're Dr. Fisher-Colbury. So I did notice they just brought out popcorn, but I seriously doubt it's going to be that exciting. So good afternoon, esteemed members of the board and public. My name is Doug Kearney. I'm the Director of Grants Management here at CIRM. And I'm here today to request approval for a modified facilities rate policy. In December of last year, the board approved the adoption of a new award management policy, which we call the AMP. While CIRM's allowable facilities cost policy was articulated in our previous grants administration policies, the AMP relies on the board to set allowable facilities rates outside of that document. This method provides flexibility to the board to make future adjustments to our facilities rate policy in an uncertain national landscape. When the award management policy goes into effect later this quarter, CIRM needs to have a policy in place. As I just described, the purpose of this proposal is to we have the opportunity to simplify and clarify the policy around allowable facilities rates for awardees and the CIRM team. Our current policy, which I will describe in a moment, has worked well for CIRM over the years and can continue to do so for the foreseeable future. But we believe some modifications may be beneficial. These modifications are limited to the allowable facilities rates for institutions that do not have a federally negotiated rate. The impact of these modifications would affect a limited number of future applicants, and I'll talk more about that in a moment. Both CIRM and the federal government provide two types of support to subsidize the underlying costs of doing research: facilities costs and administrative costs. Facilities costs cover general operating costs of the facilities, and administrative costs cover general administration expenses. We rely on federally negotiated rate agreements to establish how much total overhead our awardees can request from CIRM. Although CIRM arranges components of these negotiated rates differently by separating out facilities and administrative rates. The result of our calculation method provides an equivalent amount of funding to the federal policy. Again, we are only talking about facilities costs in this presentation. Where our policy differs from the federal policy is when no rate agreement has been negotiated. In the federal system, awardees are limited to 15% total facilities and administrative costs, until negotiations have been completed. At CIRM, we limit for-profit awardees and nonprofit awardees without established rates to 35% facilities. The 35% must be negotiated and supported by a demonstration of need, creating administrative burden for both parties when launching an award. Let's walk through our current process. So we start with an awardee. That awardee is either a for-profit or a nonprofit organization. A for-profit organization can request up to 35% facilities subject to negotiation with CIRM. A nonprofit organization either has a federally negotiated rate or they do not. When they have a federally negotiated rate, CIRM will honor that rate to establish the allowable CIRM facilities rates. If they do not have a federally negotiated rate, they can request up to 35% subject to negotiation with CIRM. Oh, and again, the items highlighted in gold are the only items that we're proposing to change in this proposal. We propose that CIRM continue to accept federally negotiated rates per our long-standing policy. For nonprofit awardees without a previous rate agreement, as well as for-profit awardees, the CIRM facilities rate would be limited to a 15% flat rate, aligning with current federal policy. Let's walk through what that process would look like. Again, we start with an awardee who's either a for-profit or a nonprofit. A for-profit applicant can request up to 15% facilities without the requirement for negotiations with CIRM. A nonprofit applicant has a current, provisional, or previously negotiated federal rate, or they do not. If the former, then we would continue to honor those federally negotiated rates. And if they don't have any of those, then they could request up to 15% facilities, again, without the need for negotiations with CIRM. Highlighting in gold the changes from the previous slide. So what's the impact of this proposal? Well, 96% of our nonprofit awardees do have federally negotiated rates, and CIRM can continue to honor those. For nonprofits without rate agreements, CIRM will still provide overhead funding, but with less administrative burden. For for-profit applicants, the majority of for-profit awardees don't request facilities from us at all, opting to maximize direct project costs within our total cost capped awards. So although this is lowering the limit from 35 to 15%, it does affect a minority of our applicants, for-profit applicants. The benefits that we see from this proposal are that it simplifies award launch and provides a clearly defined policy that more closely aligns with federal practice. It eliminates higher than average facilities rates to organizations who have not gone through a rigorous federal rate negotiation. And, you know, I skipped over something that I'm going to talk about related to that. So under our existing policy, a nonprofit without federally negotiated rates can request up to 35% facilities rates. And entities that have undergone that federal rate setting process average around 30%. So therefore, it's possible to potentially get more facilities rates without having gone through that federal rate setting process, which is something that we're hoping to correct with this. But most importantly, it retains the flexibility to adopt a new model as needed to adequately support California institutions. So the CIRM team requests the ICOC approve the allowable facilities rate structure as presented today.
So moved.
There we go. We have a movement to accept the recommendation.
Second? Yeah.
Yes, and Emery seconded. Yes, thank you. So comments from board members. We'll start with Kim Barrett. Thanks. And John.
So thank you very much for your presentation. I wondered, as of right now, it looks like the federal government's efforts to reduce F&A to 15% has been at least stymied, if hopefully retired. But what would be the impact if at some future point
Right.
California institutions are very much under supported by the federal government.
Right. So going back to the award management policy, it gives the board the authority to set our facilities rate, our overhead rate policies. So if something happened on the national scale that required a change in us relying on these federally negotiated rates, the board can ask us to come back with proposals for an alternate funding strategy.
So also thank you for this presentation. So this is only the facility piece of the FNA. So I mean, I think in the federal, it was both FNA that was going to go down. So this is only,
That's
but
correct.
I guess my question, if this is pegged at the federal rates, is the administration component, I know you didn't talk about that.
Yes.
Is that also pegged at a negotiated rate or a federal rate?
We have a indirect cost policy of 20%. So I referred that we sort of calculate things differently, but it comes out to around the same amount of funding. We're not proposing any changes to our administrative overhead rates at this time. So, you're right, I didn't talk about that in this because we're talking about facilities, but that does impact the overall overhead that our awardees can receive.
Okay. All right, John, George?
Yeah, thank you. You alluded to the fact that private entities have more flexibility with regard to direct charges. Could you say a little bit more about that?
Sure. So our award mechanisms are all total cost capped awards. So every applicant can ask for the same limit, total limit. Our nonprofit awardees generally have overhead rates that they must charge to all sponsored research projects to stay, in business. Whereas what we found with for-profit applicants is many of them will not ask us for overhead support and instead maximize up to the award cap and ask for direct project costs and not ask CIRM for those overhead costs. So that's been a very clear trajectory where nonprofits always ask for it, for-profits usually don't. No.
Okay. Any other comment from board members in the room about online? No comments. Okay. So any member of the public want to comment on the motion on the floor? And there are no hands raised. In that case, we can proceed to a vote then. Voice vote?
All those in favor say
Aye.
You got it. Thank you. And I'll poll, excuse me, those opposed say nay. Any abstentions? And I'll poll those on the phone. Judy Chow,
Aye.
Deborah Dees,
Yes.
Elena Flowers,
Yes.
Jeff Golden,
Yes.
Linda Malkus,
Yes.
Joe Panetta. And Joe has left. Carol Watson.
Yes.
Thank you very much. The motion carries.
Thank you.
Thank you.
Advancing now to item 15 of the agenda. This is a discussion on our 2023 to 24 financial audit. Our Vice President for Operations, Jen Lewis, will lead the discussion and introduce our auditor.
Thank you, Chair Imbasciani and members of the board. Thank you for giving me the opportunity to introduce the fiscal year 23-24 financial audit. As background, per Proposition 14, CIRM is required to have an independent audit of our financial statements on an annual basis, which is then reviewed and audited by the State Controller's Office for or a quality review, they call it. For 23-24 fiscal year, CIRM engaged Macias, Ginny, and O'Connell for that independent audit. And we refer to them as MGO to complete the independent audit. This audit tested the fair representation of our financial data, looked at our internal controls to ensure that we were not misrepresenting any financial data due to error or fraud. And on behalf of the CIRM's finance team, I'm happy to report that once again, there were no findings in this audit. The audit was certified by the State Controller's Office subsequent to the completion by MGO. And then in December of last year, it was presented to the Citizens Financial Accountability and Oversight Committee that is headed by the State Controller. I want to thank Director of Finance Michelle Lewis is on the line, as well as her team, Senior Finance Officer Sumi Thomason, who led that process. And now I'd like to introduce our auditors, Russell Robertson, who is the Assurance Director at MGO, and Kathleen Foster, who is the Assurance Manager at MGO, who will review the results of that audit, analysis, and any conclusions, and happy to answer any questions.
Thanks, Jill. Thanks, board, for allowing us to do a quick overview of the 23-24 audit results. Jill did a great summary of why you were having this audit. It's a requirement. And I will go through briefly some of the results and highlight some of the operations for that previous year. Next slide, please. Here's a quick agenda. I'm going to briefly go over the scope and the services and deliverables that were presented for fiscal year 23-24. Again, give you the audit results, some operational highlights, and there's I'll go into a secondary report called the required communication report that's presented to the board as well, that you should have both the independent audit report and this communication report. I'll go over that briefly and go over some of the highlights in that report as well. Next slide, please. As indicated, the scope of our services for this audit was for the basic financial statements for fiscal year 24. The audit was conducted last year and issued sometime in late summer, and now we are getting to the results. With those deliverables, we have what is called the independent audit report. In that report is the main piece of the audit where we provide our opinion over the financial statements and the material aspects of those financial statements. We also have a secondary report in there, and that report is on the internal control over financial reporting. I always like to say in this report, we assess internal control and we do test those controls. We don't provide an opinion over those, the internal controls. So you won't see that in that report. But if there were any issues that popped up through those tests that we did, we would identify those and communicate those as well. And then the secondary report that I'll be talking about is that communications at the conclusion of the audit. Next slide, please. So as Jill was indicating there, for fiscal year 24, we provided unmodified opinions over the financial statements. And what that really means in a nutshell is that the financial statements present fairly in all material respects of CIRM's financial position and the results of operations. And those are in accordance with generally accepted accounting principles. There were no material misstatements, no scope limitations. We were able to audit everything. Our focus is on immaterial areas, look at the expenditures, any of the grants that were provided to CIRM during the year, the collections of how the money is receipted. So we look at material areas in the operations of CIRM. So there were no scope limitations. We were able to test everything we could. No disagreements with management during that audit. And under governmental auditing standards, as I indicated, we tested controls and there was no findings identified. And there were no instances of non-compliance with laws and regulations. So Overall, a very clean, efficient audit, no problems, as Jim was indicating. So very easy to get through the audit, and we were able to get all the information we needed to conduct that audit. So everything was good on that front. Next slide, please. So in the next couple of slides, I'll briefly go over some very high level operational highlights, and really that was a focus of our audit. It was the revenue coming in, obviously, and the expenditures going out. So as we identified, and you'll see in the report, the change in financial position and liquidity increased by 124 million, from 213 to 337. The main driver of this was Prop 14 GO bonds, general obligation bond proceeds of 383 million, and Proposition 71 commercial paper of 52.8 million. And then there was some higher investment earnings of roughly about 9 million increase in investment earnings because of the influx of cash during the year that was held by CIRM. Next slide, please. Our focus was, of course, on the major program spending. We identify here research grants totaled nearly $300 million. That represented the majority of the expenses for the year. This was an increase of 104 million year over year from 23. We have identified the three major fund areas here for the stem cell research and cures fund. Grant expenditures increased significantly as the fund works to be fully expended, expending the Prop 71 authorized funding. The Stem Cell Research and Cures Fund, the increased payments was because of overall increased activity while new grants were awarded during the year, and some milestones were met as the completion of the grants from the prior year. And the Revenues and Royalties Fund, no activity during the year, but CIRM is working towards developing a plan, and probably for fiscal year 25, that's already rolling out, because we are a year reporting on a year and back, so that's probably already in the works, and enrolling eligible patients for the assistant awards. I believe, next slide, please. This slide focuses on that, the required communication report. In this report, this is really for you as a governing body. This really highlights any changes year over year, any new accounting policies that were initiated, any new or significant accounting estimates. There were some new accounting standards. However, they didn't have an impact on CIRM for fiscal year 24. There was a couple that didn't really materially affect the operations of CIRM. There was no misstatements as indicated earlier. If there was, it would be in this report. No disagreements as I indicated before, and no subsequent matters to report on. So overall, as year to year goes over, I mean, there was an increase of expenditures, but really a smooth transition from 23 to 24. No significant changes on how the finance team conducts operations. So very clean, and really appreciate working with CIRM's staff to get this audit completed. I think that's the last slide from my presentation, but I'm here to answer any questions if there are any.
Mark? Yeah, I just had a question. I assume that one of the steps in the process will be a review by the Citizens Financial Accountability Oversight Committee, and I think that's going to happen after this. Is that correct? Just wanted to confirm that.
This for fiscal year 23-24, the controller scheduled that meeting in late December. So actually, due to the timing, unfortunately, we had to bring that to, but that has been reviewed by the CFAOC as well in late December, and then subsequently here at the board meeting. And then next year, obviously fiscal year 24-25, we've completed that audit. We've submitted it to the state controller's office for quality review. Once we get that completed, then we will bring it to the board.
Great, thank you.
Any other questions of the auditor or of Jen? Or on Zoom? Okay.
This is Kathleen, just Kathleen Foster, sorry, just briefly. The State Controller's Office is currently in the middle of their review for 24-25's audit. So hopefully we'll be able to present it sooner time-wise from the end of the audit period.
Sure. Okay, we'll look forward to that. Thank you. Jen, a good report. It's always good news as usual. Thank you so much. Senior Director of Communications, Amy Adams is going to bring us up on update on communications, item number 16.
Thank you, members of the board, CIRM team, and members of the public listening in. I want to start this presentation. I'm always up here babbling away at you guys while my team works very hard. And I want to say right now, as I tell you about what we have been doing and will do in the future, they are working very hard getting a press release out about the RAPID program, posting a story about the board meeting so that everyone in the California public can know what's been going on. There's a RAPID blog item going up. There's 2 stories being written about the 2 presentations that happened. And all of this is happening while I stand here. I'm ignoring all the slacks that they're sending me right now. So if things are delayed, it's because of this. I think it's really important to acknowledge the people who are doing so much hard work. Okay, and with that, today I'm going to introduce a communications toolkit that I've developed to help people talk about CIRM. Developing this toolkit and training people to use it were called out as specific tactics in the strategy that I presented at the last meeting. Today, I'll also update you on a few of the activities the team has been engaged in addition to what they're doing today. As always, I start with CIRM's mission of transformative treatments and the scientists, students, and patients who are top of mind in all of our activities. And a reminder of the communications and outreach team mission, which defines our role in helping CIRM achieve its mission. At the last meeting, I presented our communications strategy. Communications and outreach strategy. It consists of 4 parts: create the story, tell the story, deliver the story, and measure the impact. The toolkit I'm going to talk about today mostly addresses the first 2 parts of that strategy: create the story and tell the story. First, highlighted in red here, the narrative and deck. That was what JT presented at the last board meeting. It is now part of the toolkit. It is available to all of you. It is posted to the agenda. You can reach out anytime and we can give you a copy of that deck with the associated script, and just teeing up something that's not quite done yet, but we're creating some variations on that deck just so that people have some different versions that might feel more appropriate for them or their audiences. Just a reminder about that narrative and deck for those of you who have forgotten it in the busy last month. It makes CIRM's mission personal. It focuses on real patients whose lives have been impacted by CIRM's work. It's also very beautiful. I think the visual element of it really helps highlight the emotions in the story. It also conveniently hits all 4 of CIRM's key messages, which I'm going to talk about next. Okay, so the next part of my strategy is telling CIRM's story. And we're going to focus there for a little bit today. Just a reminder, here's the toolkit. We're going to start by talking about some messaging. The messaging is attached to the meeting agenda, and you're all welcome to take a look at it. I'm happy to take questions now or later about everything in the messaging. I'm not going through every word of that messaging document up here because it's been a long day. I'm going to go through some key points though, just so we all kind of get our heads around how to use that messaging. To be clear, this messaging doesn't necessarily say anything new. We're the same organization with the same mission, but it helps us talk about the organization in a way that might resonate more with our various audiences. Some of you who were here might recall that in June at this very podium, I said one reason I thought CIRM was struggling to be better known in the state was because some of the words we use in talking about ourselves, institute, regenerative medicine, stem cells, these are kind of big, complex words and aren't going to resonate with everyone. This is a real word cloud made from our new messaging, and it's got some of those same words in there. You can't get away from the word stem cell and regenerative medicine, but we've got some new words in there too: therapies, cures, access, medicine, and some other words that I think will help us talk about ourselves in terms that are a little more real to people. I'll go through a few sections of the messaging today. It includes 4 key messages shown here, and later in my presentation, I'm going to give you examples of how we can use these messages. Hang on, there's an animation that is not working. That's okay. These 4 messages come down to the following concepts. Our funding focuses on cures or therapies. We're going to talk about that in a minute. We work to make sure Californians from across the entire state can benefit from our clinical trials and eventual therapies. We bring additional educational and economic benefits to the state. And we're advancing all of this to benefit Californians at a time when federal funding is less secure. And I've highlighted some key words here that kind of touch on those key messages. So really, if you use some handful of these words in a sentence, you're pretty much on message. I'm trying to make it easy for you. So the messaging document includes some talking points to help you address these key messages in conversations and presentations. I think the second page has each of these key messages and some proof points and language that you can use to talk about these messages. And I'm not going to go through them now, but I encourage you to look at them. We tried to write those in language that felt very natural, language that you could work into an email or a presentation or any piece of writing to help you be on message and also sound comfortable. Okay. The document also includes these phrases. Behind every number is a name. Behind every disease, condition, or injury is a person. We have the power to create cures that change lives. All this because Californians chose to invest in world-class science. And those are themes that were in the presentation that JT delivered. These are obviously not words that flow off the tongue, right? You're probably not going to weave this into a conversation with your neighbor, but they are themes that I think are very important and are worth keeping in mind. For the first one, anytime we talk about numbers of awards or the number of clinical trials we have funded, which these are numbers we use quite a bit, we always need to talk about the patients who stand to benefit from that work. We're not just an agency that gives out awards, we're an agency that tries to help patients. When we talk about diseases or conditions our researchers focus on, also talk about people living with those conditions. What is it like to have that condition and why do we want to help them? Focus on how your work and CIRM's activities will deliver cures or therapies and benefit Californians across the state. And remind people that California is investing in this work at a time when the funding landscape is uncertain. Okay, the final page of the messaging is called our mission in motion. The mission in motion, that's our strategic allocation framework that guides our funding programs. The SAF is how we're going to make good on the promise in our key messages. The SAF is deeply considered and thoughtfully outlined, and it's therefore a lot to take in. Which has made it hard for us to talk about it in ways that really explain its importance to the people of California. So the last page of your messaging document breaks the 6 goals of the SAF down into what I call a bite, snack, meal approach to communication. And to be clear, I don't just call it that. I didn't make that up. I think it's super smart and I borrowed it from other people. The bite is if you're not sure how much time you have or the person's appetite to learn about the SAF or about CIRM. In that case, for every goal, we have a short snapshot of what that goal means. If the person seems interested or you have a little time, you can give them the snack. And here I have the snack for goal 1. It's a full sentence. And if people are really interested, well, you know what? Our website has, I think, not just a meal. I think it has an entire buffet on the SAF. So we can go quite deep. This is just a way to help people get into concepts that might be hard to understand. but I think are incredibly important to us. Okay. So I'm going to move on to another document that is attached to the board agenda and is available to you anytime if you don't memorize the URL for the board agenda and just want to reach out to me. And that is an elevator pitch and FAQ. So at the same meeting where I stood here and talked to you about the words CIRM uses, I said I thought we needed a consistent way of talking about CIRM, a description of CIRM that all of us could use and that would help us be talking about the same organization. Because when I talk to people, when I ask people how they talk about CIRM, they might have been talking about 20 different organizations. So I got feedback. Thank you. Feedback is a gift. You will not be surprised to know that the feedback I got depended on who it came from. People had very, very different ideas of what was important about CIRM. And from that, I realized there's never going to be a description of CIRM that all of us can use and all of us will feel comfortable with, and that will be appropriate for all of our audiences. So this is written as elevator pitch in the singular, and it's really elevator pitch in the plural. And it's sort of a choose your own adventure elevator pitch. So I'm going to go through that a little bit so you can think about how to use it. Okay, so the first, oh, this animation is working. All right, so here's a proposed sentence that you could use. You're talking to someone you don't know. So you're at a dinner party, you've got no idea who this person is. So a good sentence might be, CIRM is a California agency that funds research intended to produce therapies for people with incurable diseases. And I've highlighted the words that mirror our messaging. So whatever questions the person comes back with, they're probably going to be questions that are on message, and it will allow you to talk about things that are critical for CIRM. Okay, and I'm going to give you a longer example. And this is an actual script that I use. And I know you're not supposed to read a lot of text on a slide out loud, but I'm going to break that rule only because I want you to hear what it sounds like conversationally. So this is actually what I would say. CIRM is a California agency that funds research intended to produce therapies for people with incurable diseases. You might remember that in 2004, there was a proposition to fund stem cell research in the state. That's how we were created. In 2020, another proposition extended our funding to include gene therapy and to include a focus on neurological diseases. We fund science from research breakthroughs through clinical trials. We train students to work in regenerative medicine labs and make sure Californians all over the state can access our clinical trials. Okay, this one works for me, and here's why. I'm old. So a lot of the people I talk to, they remember Prop 71. And so if you can jog someone's memory, you can kind of pull them along from there to talk about who we are now. Okay, I was introduced to a friend's kid the other day who just graduated from grad school in the field of science, but she's not as old as me and has probably never voted. So I didn't use this. I used a variant of this, but without the propositions because it wasn't going to resonate for her. So this is one example built on our messaging. It is not a mandate, but I hope it's helpful to people. Oh, and here's my animation working where I highlight, here's our key messages embedded within that elevator pitch. All right, let's go back to the bites. How do you take that first bite, the unknown audience, and say, okay, I'm pretty sure I'm at a dinner party with a bunch of scientists. I don't know exactly what kind, but maybe I'll work in CIRM is a California agency that funds stem cell and gene therapy research intended to produce therapies. That might be information that's useful to a lot of audiences. Or this one, you can see in red, I added from discovery research through clinical trials. If you're somewhere where you think you probably are talking to someone who knows a little bit about biomedical research, they might be interested to know it's discovery through clinical trials because that's not what everyone does. And I want to give another example, and this is Maria Bonneville delivered right over there. And I'm going to say something good, Maria, so you want to listen. Maria delivered a masterclass the other day in how to use our messaging. It was in a media interview. And she was on the interview to talk about the CCCEs and more generally about access. Maria is the head of the working group for access and affordability. So that was her role on this call. So she said, CIRM is a California agency that funds research intended to produce therapies And then she pulled a line from the messaging and said, and put those therapies within reach of people in California, which tees up access and affordability. And I didn't actually jump up and hug her in the interview because I was on Zoom. But after, it was a perfect way of taking our messaging and mixing it up for an appropriate audience, but staying right on message. And she teed up the conversation perfectly. Shyam Patel, if he's still in the room, was on that call too. And yeah, I think they did a terrific job together. Yay. Okay. All right, I'm not going to talk about this in the presentation, but that elevator pitch document also has some FAQs. These are very light, very high level. They're the kinds of FAQs that anyone might get in a fairly casual setting. They are not intended to get you through an entire conversation. I can't help you with a whole dinner party, but I can probably get you through the first couple questions that come up in a conversation in a way that sort of keeps you on message and keeps the level right. So please read those FAQs. They're the first draft. I don't think they probably are complete. There's some questions that you're going to get that I don't know about. So please reach out to me. Please give me feedback if you use my FAQs and it goes horribly awry. I want to know about that. Okay, now I'm gonna catch up with my script to make sure I said everything I wanted to say. Okay, so another part of the toolkit, and this is something that is not complete. So I can't give this to you now, but it will be complete. So I want to talk to you about it a little bit. So we're calling this a patient and student story compendium. So we have lots and lots and lots and lots of stories about various patients, and we have lots of stories about our incredible students. They're a little bit buried, and they're hard for the people of CIRM to talk about if we don't surface them for you. So we're going to have a series of, I believe there'll be slides. Each slide will have a picture of that patient or that person. And some information, the person's name, the disease, details about the trial, links to award information, links to a blog entry we've written, prevalence of that disease, different ethnic groups that might be more impacted by that disease. We'll put whatever information in there we have. You can pull that out, and that allows you to use these stories and these slides in whatever context you want. Maybe you don't really want to talk about that particular patient. You just want an image that represents a field of research you're going to be talking about. That's fine. We've got a slide for you. So this is coming. And then finally, I mentioned numbers. Behind every number, there's a name. Well, we use a lot of numbers. And in my first, I think I'm on 6 months here at CIRM, I will tell you, we use very different numbers. They're different in different places. So one of the things I've been doing, and I want to say I'm working very closely with people on Rose's team who are exceptionally picky. And we're trying to pull together numbers in a way that we're actually using the same ones. We'll have dates on them so that if you want to give a presentation and you want some numbers about CIRM, you've got it at your disposal. You don't have to email us. We'll be starting with numbers that we have at our disposal and then hoping to expand what we can provide. So look for this. I tell you, if we get this one done, that's blood, sweat, and tears. Okay, and then the rollout. This is a happy photo of some of our board members, and then Jacqueline Hankin, who does outreach for us in Southern California, at an event. This is to make the point. All of this is available to you to speak about CIRM, which tees me up to say, I've been warning you of this, and we want board members to get out and talk about CIRM, and we're going to make it easy for you. Aditi Desai on my team is tasked with helping board members find opportunities to speak. She will now be reaching out to you. Over time, we'll figure out what kinds of opportunities people like, what you don't, what are appropriate opportunities for each of you. But I think we can do a better job of getting all of us out talking more. And we will try to make it easy for you. Okay, and a presentation from communications would be incomplete if I didn't talk a little bit about what we've actually been doing, because all of this has been about what we're trying to do in the future, and we're kind of tired of looking towards the future. What are we really doing? here's some of the stuff we're doing. I'm mostly going to talk about outreach today. We do other things, but I'm just really excited about some of this. So our community outreach team will be at a student-focused event this weekend, and they will be accompanied by 3 of our education program students. Maria Bonneville and Aditi spoke at a Rotary Club meeting. I think that was last month. Jacqueline, who assists us in Southern California, represented CIRM at an ALS summit recently, which attracted about 200 people. She reports that the program featured a dedicated page recognizing the importance of CIRM to the ALS network. In addition, Aditi recently spoke to a Sacramento area Rotary Club where she gave the presentation that JT previewed at the last meeting. So we're starting to roll that out, and she said it was very well received. Participants had questions about access, specific disease areas, and long-term implications of cell and gene therapy. So they were very engaged. We've done more, but I'm going to leave it there on this very long day. Measure the impact. I'm not prepared to give you a full metrics report today, but I'm going to bring in one of my team members at an upcoming meeting and talk in much more detail about metrics, because I think it's incredibly important. The one thing I'll touch on lightly here is, I have images here. At previous meetings I've mentioned, we've started doing blog entries on our board members. And we're doing that as a way of humanizing what can look like kind of a bureaucracy. These have been doing incredibly well. People seem to really resonate with these. And the other is a blog entry we did a while ago on our education programs over the past 20 years. And it continues to do well. I think those programs really resonate with people. And while I have the floor, thank you. And right before I take questions, I want to say, earlier today, someone came up and asked me about social media strategy, and how people, if you're comfortable on social media, could amplify CIRM. And I will just say that in the next week or 2, those Closer to Cures presentations will be on our LinkedIn. So if any of you are LinkedIn users and want to look for them, I'm not insisting that people promote them, but if you liked the talks, we would not mind it if you said something. And with that, I'm going to come back to something that I promised I was going to tee up. And that was at the last communications subcommittee meeting. We had an outstanding conversation, I thought, about some of the words we use to talk about therapies or cures or maybe treatments, some combination of those, and just how we're talking about what it is that we're doing. You'll notice in a lot of my work, I'm using the word cures. That's because it's short, and a lot of what I do is short. I think in some ways, cures also is an important point in regenerative medicine. It is a field of medicine where we could hope for cures, and we want to lean into that. In other contexts, I know from talking to all of you, it's not a comfortable word to use, and I know that. My takeaway from the conversation was that I think we all really need to think about our words and use words that are relevant in our contexts. I don't have a mandate, and I'm not going to tell people which word they have to use. I kind of trust all you to be grown-ups and choose your words. But I would welcome more thoughts on that topic now or any other time. And with that, I'm done. Thank you.
Thank you, Amy. Any further comments? Question? Yes, Yael?
I'm sure I should know this, but where does the toolkit live so that we can reference it?
Well, right now it lives attached to the board agenda, but in a slightly draft form. You can grab it, but there's little updates along the way that people have commented on. So I will be at some point sending you guys a packet that has the final. You're welcome to reach out to me anytime also. One thing I think I forgot to mention is I am going to be holding some trainings on the toolkit in February. They'll be for staff, but we'll invite, I'm looking at Scott now, we'll invite the board in a way that is okay according to Bagley-Keene, and Scott is going to make that possible. So we'll be talking about how to use the deck, how to use some of the images that we've been developing. So we'll do that as well.
So I was wondering if we do communications and tell our story in languages other than English?
Oh, well, okay, you beat Isabel to that question. So congratulations. So I'll answer to both of you. Yeah, I think that's an outstanding question. And right now, I will say we have no materials to help you. I think it's an important question though, and I think it's a place where we need to focus in the future. We are starting to try to get things like press releases translated, and we're reaching out to Spanish-speaking media, but it's limited at this time. But I think other language, we're a diverse state. That's not the only language in the state. So at this point, I can't help you, but I'm happy to have a conversation if that's useful to you. We have Marie makes a good point. We have annual reports in Spanish. We also have a series of patient-focused flyers. And someone on my team is going to Slack me in just a second if I say this wrong, but I'm pretty sure several of those are also translated into Spanish. Okay, Maria says yes. Okay. Yeah, so we have some translation.
Thank you very much. Excellent presentation that will help us talk more about CIRM in our daily life or so. A general question, not necessarily just for you. When we're asked the question, how much fund is there still to be allocated? From now on, I'm told 3.1. I just wanted to make sure that we all have the same response there. 3.1
Okay.
billion.
That is a number, I'm looking at Jen right now. That is a number we're currently updating once a year in the annual report, and it will be in the proof points document with a date on it. And Jen, is that right? Are we doing that once a year? Yeah. Okay.
But
when we're doing the reverse, we're talking about how much has been spent, not how much is left.
we want how much is still to be allocated?
Yes. Okay, is it on that? Okay. So 3.4 billion as the start of this year is what we have for the next 6 years. But that is a number we will report on every year as we come with a new budget. So and then we'll make sure that's in your materials, so you have that as well.
Great, thank you.
Thanks, Jen.
Amy, that was a great presentation. And in reference to the translation of products, being on the grants working group, virtually every grant that goes to a clinical trial, the investigators say, we are translating our instruments, our call for study. I'm imagining some of that material might be useful to us. Can we access that? Have you thought about that?
Yeah, okay, so I'm going to dig back in time now to my last stint at CIRM. I would say right now, not so much. Is it available and should it be used in the future? I think it's worth mining for sure. In the past, we made the public summaries, like we did things with them. And then oh, is that different?
Yeah.
Okay.
When they talk about access to the trials and population impact.
Okay, sorry. I was
they
thinking
talk
about the public.
Yeah.
I'm sorry. I was responding about the public statement. And no, actually, I think I'm pretty sure that right now we do nothing with that. And that's
Wondering if our alpha clinics are preparing documents they're using to educate patients in different languages. We might be able to access those as well as a starting point anyway.
I think the alpha, just as a general statement, I think the alpha clinics are doing lots of amazing things to get the word out, and it's something that we're wanting to expand. Yeah.
Mr. Chair,
Yes.
you have a left bias.
Bias, right?
Isabel. Actually, I'm going to be doing something with the Oakland Rotary. And what I always suggest is, depending on where you're presenting, you think locally. So my immediate questions to Amy and put the team to work was, how many bridge programs do we have in this area that we can point at? How many companies in this area who have gotten CIRM funding and are doing what? So that we can point to the activities within their sphere of knowledge and influence and saying, oh, we are getting something out of this. This is building jobs, building teams of scientists going forward. I think that's always critical. What is it that politics is local? And so I think it's really critical and I know that creates more work.
My team is happy to do the work for you, Isabel.
In looking for the data, but I always believe that that's kind of, and what about me response? And so and so I would suggest that anytime they ask you to speak, ask them to give you that localized impact that's already happened within their communities.
Okay, Isabel, anyone else? If not, I'm going to thank you, Amy, for a great presentation.
Thank you, everyone.
So we skipped around a little bit to accommodate various exigencies in the schedule. We're going to go back now to item number 9, is the report from the Vice Chair. Maria?
As many of you have heard, NIH is issuing a request for information on reducing reliance on human embryonic stem cells in NIH-supported research. And while they await these responses, HHS is pausing new submissions to the NIH Human Embryonic Stem Cell Registry. And our teams have been in contact with some of our partners, ISSCR, ASGCT, and in addition, that team is already working on a plan to coordinate a response to the RFI with our California institutions. And so I'll keep you posted and updated as things unfold, because that's a new big development. And in addition to that, there was also news out of NIH that they will stop funding research involving fetal tissue. And I've had a lot of questions on how this will affect CIRM. And the short answer is it doesn't, because we will continue to fund this work. It will, of course, affect some of our partners. And so we'll also monitor the situation and provide any updates as we get them. The Senate Help Committee is holding a hearing on February 3rd on modernizing the National Institutes of Health, faster discoveries, more cures. And the MAHA Institute is hosting an event on Friday on Reclaiming Science, the People's NIH, featuring presentations from NIH leadership. And based on the agenda items, it looks like it's focused on NIH reorganization, criticism of COVID responses, gold standard science, et cetera. And so as soon as we hear back on how those go, I'll send something out to the board. We've been tracking Senator Wiener's bill, and that was resubmitted and is now SB 895. And that's a $23 billion bond measure. And Wiener has asked the legislature to put it on the November ballot so it avoids signature collection, which, as we know, can be very costly. And it covers a wide range of scientific research, including biomedical, new and emerging health threats, disease prevention, stroke, heart disease, child and adult leukemia, infectious disease and HIV/AIDS, wildfire prevention, promotion of healthy and safe behaviors, communities and environments, addiction and substance abuse, behavioral, climate, weather and oceans, coastal and marine ecosystems, agriculture and water, emerging technologies, safety, efficacy, and security of drugs. So it's a broad reach. The bill is currently sitting in the Senate Rules Committee pending referral to the Senate Health Committee. The Health Committee will begin to hear newly introduced Senate bills in mid to late March. If it's heard and approved by the Health Committee, the bill would be referred to the Appropriations hearing for consideration. If approved by the Appropriations Committee, the bill will move to the Senate floor for approval by the full House. Once approved by the Senate, the measure will go to the Assembly, where it will be heard in the Assembly Health and Appropriations Committee before going to the Assembly floor for a full vote. If it's amended in the Assembly, it has to go back to the Senate for a concurrence vote. And then before that goes to the governor for signature to be placed on the ballot. And that has to happen by around June 25th, unless that date is extended, but no later than July 2nd when they recess. So that's where that stands. So there's a lot of hurdles between now and then. And then there's also a potential bond measure focusing on immunology. And this proposed bond measure is currently out in the field for signatures and must qualify by the end of June in order to make it to the November ballot. And it proposes $8.4 billion in bonds to fund cutting-edge medical research focused on immunology and immune therapy. About half the funding would go to one major nonprofit research institute affiliated with the University of California. The other half would be awarded as competitive grants to California public and nonprofit universities and research institutions. And at least $4.2 billion must be spent specifically on cancer, heart disease, and Alzheimer's disease research. And that is my report, if you have any questions.
I see a hand. Yeah, it's George.
Thank you, Maria, for that comprehensive report.
Sure.
A couple of quick questions about governance.
Sure.
I mean, for both the Wiener bill and the other bill that you mentioned,
Yeah.
what mechanisms are being set up or being considered in order to disperse funds? Is there an equivalent of the ICOC?
I'd have to go back to the immunology for the senator's bill that's going through right now. Let me just bring that up. It does establish the California Foundation for Science and Health Research Foundation, and that lives under the Government Operations Agency. And their role is to facilitate the scientific research by awarding grants and making loans to public or private research companies, universities, institutes, et cetera. It's not completely clear how the oversight is handled in that instance. What I do know is that they did go back and expand or add language to the bill that has a lot of similarities with CIRM in some of the instances around closed session and otherwise. So I can get back to you with more information on that. Yeah.
Any other questions for Maria? Any other board members online? No. Okay. Great. Our CEO and President, John Thomas, President's report is next.
Last and probably least, but maybe not, wanted to give you an update on a number of things. Two, very briefly on events that have happened, two on things that are going to happen, and one on a presentation which I've streamlined and have called out some interesting facts that I think you guys will like as you head out into the night after today's very busy session. So, with respect to things that have happened so far, as Jen noted, on December 30th, the CFAOC met to, as it does annually, to hear the story of our audit, to hear the story of our budget. This was a virtual meeting. Maria, Michelle Lewis, and I did the call for CIRM when it went very well. In the past, we've had discussions about the cool things that we're doing, and boy, did we hear some today. And this particular instance, given the late day in the year, we did not get into that, but presumably you'll be reconvening with the CFAOC sometime in the coming months to give them an update on that. Vito alluded to an annual event that precedes JP Morgan Week. The ISSCR sponsors something called the Nucleus Forum, which is a very unique combination of academia, industry, and finance that convenes in Palo Alto for a keynote fireside chat on the Saturday night, and then a full day of panels. This entity has been around 10 years. It's put together by CIRM grantee Deepak Srivastava, Harvard Med School Dean George Daly, and Harvard researcher Len Zahn, for those of you who may know the two of them. And it pulls together every year a really interesting group of people. That the fireside chat this time was the guy at Meta in charge of healthcare AI, which was interesting, although just as CIRM has and life science has its own set of impenetrable acronyms, so does AI. So just when you thought you knew what he was talking about, he'd sort of lapse into some acronym. You sort of got a bit lost, but nonetheless, it was a very entertaining talk. The panels were variously in academia, industry leaders, and finance. The principal benefit of this, Vito and I and Maria were there this time, is always the networking that you're able to do. There are always new people there that can form a very good group of people to know to help advance CIRM's agenda on behalf of the people of California. So those were two events that have happened. Coming up, you may recall in October, the board approved the CCCE delivery sites, one in Central California, one in the Inland Empire, and one in Southern California. Conspicuously missing was one in Northern California. And I just want to let the board know that we've been doing a lot of reconnaissance and intelligence on potential sites in the northern part of the state through a variety of conversations with stakeholders in the know. And we are going to be coming back to you in March with a concept plan for the selection of that final site to round out the geographic diversity of the program. Similarly, just wanted to let you know further down the road on the subject of alpha clinics, been discussions in various fronts in connection with that. The CIRM team has been evaluating how the programs are going around the state. And at the appropriate time later in the year, we'll be coming back to the board with recommendations for next steps with in connection with alpha clinics. So that concludes my very brief run through of the 4 items. Now, if I might, so over the course of the year, I've asked at various times about, and others here are as well, about the state of the field, where the development of CGT in industry, the international competitive landscape, the regulatory environment, all that stuff. And as it happens, every year, that and more is always encapsulated in an extremely highly attended speech and panel given by the Alliance for Regenerative Medicine, the speech given by Tim Hunt, the CEO, and it covers a wide range of topics. I've reduced their slides to a manageable amount, and in the interest of getting everybody out of here even sooner, I'm going to reduce it even further to score points with all of you. So the sort of the bookend story that started Tim's speech was about patients that demonstrate the potential for cell and gene therapy. You've heard a lot, particularly from Sham today, about baby KJ and what happened with him. Also, they talked about the woman, Marcy McHugh, who is the first person ever to get CAR T clinical trial treatment for multiple sclerosis, demonstrating the branching out effect of what CAR T treatments can reach to. I'm going to skip over this because we don't need to get through this now. This is interesting and frankly disturbing. Global competition, and we were talking, we had a sidebar over here about this very issue. If you'll notice here in the CGT space, the US, which has always sort of led the field, is starting to lose some ground in what could unfortunately be the start of a trend. And you see that China made very significant strides over the course of the last year, largely due to their streamlined regulatory process, not to mention all the money they're plowing into the regenerative medicine space. The Middle East is becoming more and more of a factor as they look to develop the biotech industry and increase the role of that area of the world in that field. And the EU passed an act which was meant to accelerate clinical trials. It created an advanced therapy medical product center of excellence. It extended patent rights and provides 10 billion euro to jumpstart a bunch of companies. So things are happening in other parts of the world, and if we're not careful, we're going to sort of progressively find ourselves with more competition at a minimum and falling behind in some areas at worst. Okay, now this is interesting. So baby KJ hit early in 2025, and it what it did was it generated, when the new administration was starting and the new HHS hierarchy was in place, they're looking for wins. And this was like a really early win. And it led to a most unusual event, which was in March. They convened 27 stakeholders in the cell and gene therapy space. at a site in DC to give basically testimony, and this was researchers, patient advocates, industry, et cetera, give testimony to members of the new HHS team about the benefits of cell and gene therapy. Included in the 27 were Crystal Mackel, interestingly, Don Cohn, and a number of other names that you all would recognize. It was convened over by the new head of the FDA and the new head of CBER. And they went through all 20, how many here happened to watch this? It was on YouTube. A number of people watched. Well, it was interesting, to say the least. All the things that were said were great. About 45 minutes before the end of it, they took a break, and the two of them were joined by RFK Jr., by Dr. Oz, and by Dr. Bhattacharya. So the five now, everybody involved, the leaders of healthcare, were listening to what's going on in the field. That led to a number of sidebar, as I've been told, sidebar discussions that demonstrated interest in cell and gene therapy by the leadership there. And you can see on this slide a number of the things they're doing to make it easier to get things through the FDA in our field. And you see those on the right. I won't go through them. The secretary himself is a fan of cell and gene therapy because it deals with treating the cures as opposed to chronic diseases, which is what he has a little bit less focus on. But this is one of the things, the regulatory flexibility in CMC. You can see Marty McHarry's comment, and the head of CBER, acting director of CBER, et cetera. So that was interesting. Now, this is would have been a great entree to Sham had I gone before him, but going behind him, this demonstrates the interest that he outlined in the in platform approaches to treating rare disease. And it's not just us, but in Europe, they're very interested in the same thing. So I think you're going to start seeing that as an area of major emphasis. And I'm very proud to say that I believe CIRM, with our RAPID program, is going to be on the forefront of developments in that area. This is you might find interesting as well. This is a scorecard on how the Office of Therapeutic Products at the FDA is treating companies that have products that are coming up to market. And you can see some of them have PDUFA dates. Some of them have complete response letters, which, as you know, are documents which basically said you got more work to do and defining what you have to do to come back to them. And various BLA referenced items here. So there are a number of things coming up. ARM, this is a scorecard that ARM has put together, so they're keeping track of what's happening in the industry and can update everybody on how the FDA is handling things. This is, I think, pretty interesting. So Big Pharma is progressively more and more getting into the cell and gene therapy space. I will say it took a while. But as you can see here, 20 of the 30 largest now have either individual products or, in many cases, multiple products that they're pursuing. And I think that that's a great sign for the field. It's a great sign for, among other things, our efforts to try to help match our awardees with industry in one fashion or another. And so you saw that by at this Nucleus Forum I referenced earlier. If you looked at the panel of who's there on industry, it's the EVP of product development at Amgen. It's people from Lilly, people from J&J. So a lot of the companies represented here are in that group. Now, this is the financial slide, which shows that in the past 4 years, up to 21 through 24, you had 2 blockbusters, which are defined as $1 billion in revenue, one of which Zolgensma in SMA with Novartis, and you had Yescarta, which is, of course is Kite's CAR T product in large B-cell lymphoma. And then you look and they project 2 more came into the mix last year, which, as you can see, are Carvykti, which is a multiple myeloma product by Janssen, and Breyanzi, which is another CAR T product in large B-cell lymphoma by BMS. And then you can see what they expect over the course of the next few years. So the field is in terms of big winners. I mean, this is not many, but it's a start, and it's improving year over year, and it is evidence of the field further maturing. And you can see there at the bottom, 10 total blockbusters expected the next 5 years. This is some, if you're wondering who's investing in the sector. So we had 11.1 billion last year, and you can see how that breaks down between venture, equity, debt, et cetera. A number of very big ticket M&A and partnerships. Again, you can just read the slide there. The investors making venture financings in individual companies. You can see here on this list a number of different players in the field and what they got over the course of last year. This is kind of interesting. You wouldn't have thought this was necessarily the case, that our sector comprised a higher percentage of biotech venture last year than the year before, which is a good sign further. Then you've got public offerings, of which there were quite a few, of 100 million or more, which is noteworthy because the public offering market in general was way down last year. You'll notice the top right 2, Capricorn and Tenaya, and the bottom left 4DMT. Those are 3 of the companies that we have helped fund over the years. So this is evidence of CIRM players getting into the game in big ticket financial manner. Then you have, this is sort of what they expect coming up here. So in 2026, they expect 4 more companies making it to market in the US, 2 in Europe, and a number of other indications that are working their way up and getting close to the finish line, which you can see on the right-hand side there. Potential clinical milestones that advance the sector. I'm not going to go through these. By the way, this is all on the slide on the agenda. I welcome you to take a look at that. They sort of end by saying, I missed that. They had a litany of reasons why they felt that this demonstrates that the sector is further developing. Oh, thank you. Very nice. Thank you very much. Which you can just read here. Science is great, patient impact undeniable, competition increasing, commercial opportunity and investment increasing, strategic biopharma relationships increasing. Don't want to say that this is this depicts something that's like anywhere near what this is going to be in 5 to 10 years. It's again just getting started. The financial atmosphere continues to be tough in life science, as you all know, but there are indications here that things are continuing apace. Tim Hunt sort of ended by saying that this is very reminiscent, those of you who track the development of monoclonal antibodies and that industry when it first got it started, with the challenges it faced and the sort of midlife off and on developments as the field matured, and now well into 30 years later, it's a $250 billion market. And there's every indication that at the end of the day, this is going to end up something similar. So thought those numbers would be interesting for people. And with that, Thank you very much. And any questions, happy to answer.
Thank you, JT. Great analysis of the landscape. Any comments or questions from board members?
Thank you, Mr. Chair.
Thank you. Thank you, Mr. President. I have one item that we passed over. I need to take care of it. It takes your vote.
We're
It's
just gonna punt to March,
We will, the consent agenda?
the min, it's just the minutes.
It's just the minutes. Okay, fine. We're happy to do that. So, is there any member of the public that would like to make any comment on any item on today's agenda or on any item that was not on today's agenda? Are you seeing any hands? No, I'm not. Okay, then I can adjourn the meeting. We will next convene at 9 o'clock in the morning on Thursday, March 26th at the Westin Hotel in Sacramento. Okay, good.
And I just wanted to thank board governance for a really great meeting. So thank you. And for the Closer to Cures. Thanks for introducing that and making it a great addition to our
Yes.
board meeting.
Thank you, everyone, for being here.